Our mission is to make lives better for the Filipino. In more than 3 decades, the Philippine Council for Health Research and Development has invested in research and innovation, in people, in infrastructure, and in support systems to create an enabling environment for health research in the country, to generate innovations and solutions to our pressing health problems.
Created in 1982 as a sectoral council of the Department of Science and Technology, the PCHRD is mandated to coordinate all health research in the country. The PCHRD has grown and expanded its services in the areas of agenda setting, research management, capacity building, research dissemination, translation and utilization, resource mobilization, and national and international collaborations. The PCHRD promotes health research ethics as the country's commitment to the protection of the rights, welfare, and well-being of human participants in research. Generated from the funded projects of the Council were important healthcare policies, products, and services. Even before the passage of the Technology Transfer Law of 2009, the Council already undertook technology commercialization, which seeded the growth of medicinal plant products in the country. Lagundi for cough and Sambonga's treatment for kidney stones are now household names. With health innovations in the pipeline, the Council is, at the same time, building the capacity of research institutions and researchers to make sure that research projects are technology and industry ready. Research for evidence-informed policy making is an equally important path for our research outputs. Our supported researches contributed to the development of the expanded program on immunization, the Newborn Screening Act, the National Health Insurance Act, the Philippine National Health Research System Act, the National Integrated Cancer Control Act, and the Universal Healthcare Act. To ensure that the country has a cadre of researchers who will undertake needed researches, the PCHRD provides scholarships, research fellowships, and other assistance for capacity building. What sustains a health research system is communication. With this in mind, the Council maximizes the use of traditional and new media. Partnership is employed as a strategic approach. Partnership with media, partnership with the regional consortium's communication teams, and partnership with everyone who shares the Council's vision. Decades may have passed, but the PCHRD's mandate remains the same. PCHRD, making your life better.
Gumanda ang buhay ng Pinoy. Yan ang layunin ng Philippine Council for Health Research and Development. Sangay ng DOST na katuwang ng mga Pinoy researcher, dama natin ang PCHRD sa mga tuhod na may bagong lakas ng Axis Knee System, sa herbal na gamot at supplement na nagpapabuti ng kalusugan, at test kits na gawang Pinoy. Kaya naging abot kaya.
Patuloy ang research, patuloy ang suporta, patuloy ang PCHRD.
For the very first time, in March 2023, the Philippine Council for Health Research and Development, PCHRD, will recognize outstanding individuals and institutions who became part of its journey in making Filipino lives better through health research and development. In the biggest health research event, the PCHRD Heart Awards. Outstanding Technical Evaluator Award recognizes the outstanding and invaluable contribution of technical evaluators in providing technical and expert advice and recommendations needed by the PCHRD during appraisal of proposals until the closing of projects. Outstanding Technical Working Group. This award is given to an assembly of experts who strongly work together in developing procedures and other significant activities necessary for the research program area to meet a specific objective. Outstanding Technical Advisory Group. This award is given to the Technical Advisory Group who provided invaluable contributions in effectively guiding the Council in accomplishing the objectives and deliverables of its various programs towards the achievement of its vision. Outstanding Health Research and Development Consortium Award. This award is given to the Outstanding Regional Health Research and Development Consortium in recognition of its remarkable commitment, efforts, and accomplishments in transforming the regional health research community through health R&D. Consistent coverage of Palakayang Heartbeat. The award will recognize media partners who constantly participate in the Council's press conferences and are able to publish news releases and other publications about PCHRD. Outstanding Communicator in Health Research. The award will recognize media partners who helped us bridge the gap, making PCHRD-related health research information and technical details relatable to the general public. Media Institution of the Year. The award will be given to the media institution who feature the most number of publications, news releases, interviews, and videos combined related to PCHRD and its supported projects and initiatives. The winners will be awarded this trophy, designed by the famous sculptor, Mr. Juan Sajid Imao. The trophy symbolizes the continuing commitment and the collective effort to address healthcare burdens and promote better health for the Filipino people. It shows how solidarity and cooperation in health research can help achieve our mission of making lives better. A symbol of Filipinos, uplifting Filipinos.
Over the years, the medical community has come to realize that improvement in healthcare could not be possible with only patient management and clinical care. By bridging the gap between healthcare practices and new developments in research, progress in the country is imminent. It is with this purpose that the sole and pioneering MD-PhD in Molecular Medicine program was birthed.
The program aims to improve the Philippine healthcare standard through research by producing a hybrid class of doctors, the physician scientists. It is envisioned that a medical doctor with a strong background in basic and clinical research is a more effective professional who can keep up and also contribute to the progress of the different fields of medicine.
The UP Board of Regents approved the program in 1999, and through partnership with the Department of Science and Technology and the Philippine Council for Health Research and Development, the first batch of DOST MD-PhD scholars was welcomed in 2010. DOST PCHRD offers full scholarship to the students through their 8-year program, which is composed of 5-year medicine courses and 3 years of PhD units.
The program envisions to produce graduates who will spearhead basic and biomedical researches, generating innovative ideas in the different fields with relevant impact on national development.
Admission to the program is done by selecting from the pool of qualifiers to the UP College of Medicine. Candidates for interview are evaluated with the following criteria: graduates from a biomedical field, preferably in biochemistry, molecular biology, or biotechnology, with a general weighted average of at least 1.75, and students with high aptitude and motivation for advanced study in molecular medicine and related areas in the health sciences.
The MD-PhD Molecular Medicine curriculum trains students in the field of biochemistry and molecular biology, medical microbiology, and medical parasitology. Said fields comprise the core courses, which also include epidemiology and biostatistics. These PhD courses are taken in the first year of the program. To have a holistic training in molecular medicine, theory is complemented by laboratory courses and by research electives that are taken with the medical courses.
Since its implementation in 2010, the students and graduates have continuously worked on several research endeavors. By 2021, the college has accumulated a total of 131 local and international publications. Basic and clinical researches produced by both students and graduates have been awarded in both local and international conferences. Even in the midst of the pandemic, the MD-PhD members, with funding from DOST PCHRD, remained steadfast in taking part in numerous research activities. Research on therapeutic options against COVID-19, such as tocilizumab, VCO, and convalescent plasma, immune and genetic markers for prognostication, cell-based immunoassay for diagnosis, and efforts looking at the preparedness of the healthcare and education sectors during the COVID-19 pandemic. To cultivate the spirit of camaraderie, social functions and team meetings are held to open up talks among students and the committee. This helps in improving the curriculum while strengthening bonds amongst different stakeholders of the program. Through the years, the UP College of Medicine works relentlessly with DOST PCHRD to prepare the future physician scientists by updating skills and knowledge through research electives, conferences, trainings, and workshops within and outside of the country. To inculcate critical thinking early on, first-year students perform individual biomedical researches using different approaches and technologies. During their dissertation, MD-PhD candidates can work in laboratories within the country or partner with different institutions abroad through the MD-PhD Sandwich Program.
My dissertation project is about the effect of a certain plant against psoriasis. And its implication is, of course, if it is proven to be effective, it will surely help people with psoriasis and patients with skin diseases to alleviate their skin problems.
So for my dissertation, I developed an organ on a chip that mimics female reproductive tract during pregnancy. So I use it as a model to study ascending infections affecting pregnant patients.
MD-PhD program helped me to think outside the box, and it helped me to think more about whys and hows.
This program helped me understand medicine from the molecular up to the patient and even up to the population level. It also gave me the opportunity to help my patients not just by direct patient care, but also by doing health research that generate data and evidence that can be used to improve clinical practice and even policies. And I guess during this pandemic, we saw the importance of this skill that we have as physician scientists because we can use molecular health research as a tool to fight this pandemic.
The MD-PhD program provided me with lifelong mentors who helped me from my training until now in my professional life.
I trained in a university where every clinical department has an MD PhD or a PhD faculty. And to me, that is very strategic because clinicians can work hand in hand with scientists in tackling health research questions and answering clinical dilemmas. I was also encouraged by how easy it is to collaborate with professors from other departments. And I also want to have that collaborative and vibrant research environment here in the Philippines, where health research is not monopolized by clinicians, but rather an interdisciplinary collaborations between biomedical scientists, engineers, and even social scientists.
The training abroad exposed me to the advances in science outside the country, and it gave me a vision of how the Philippines can still grow in the area of biomedical research.
To sustain this effort, programs such as the Multi-Omics Research.
All right, so we encourage our young graduates to join the MD PhD program and apply at the UP College of Medicine. Isang mapagpalang hapon po sa inyong lahat and welcome to the oral research paper presentation competition for the grantees of the DOST PCHRD Undergraduate Thesis Grant in Natural Products. I am Dr. Giancarlo Malabad, an assistant scientist at the Philippine Council for Health Research and Development of the Department of Science and Technology. And I am very honored to serve as your moderator for this afternoon's event. Just to give a background for everyone, every second week of August, the health research community celebrates the Philippine National Health Research System, or PNHRS Week. The theme for this year's celebration of the 15th PNHRS Week is Health Research, Responding to Challenges Towards National Recovery and Resiliency. One of the activities in this year's celebration is the oral research paper presentation competition for the grantees of the DOST PCHRD Undergraduate Thesis Grant in Natural Products. This competition aims to showcase and recognize the selected grantees' innovative work on natural products. So this is going to be a very exciting and interesting afternoon for all of us. But before we begin, may we request everyone to please rise for the singing of the Philippine National Anthem, to be followed by a short prayer.
Let's bow our heads and place ourselves in the holy presence of our Lord. Heavenly Father, we thank you for gathering all of us today in this important event. May you bestow us your wisdom and guidance so that we can appreciate its significance in our lives. Bless everyone present here today, that each of us may be able to share what we have for your glory and honor. Always grant good health and safety and peace and goodwill to all men. May all the various health research activities of the Council and all our implementing agencies be a success through your interventions. We ask all of these in your name. Amen.
To formally welcome us to this event, may we call on the Executive Director 3 of the Philippine Council for Health Research and Development, Dr. Jaime C. Montoya, for his opening remarks.
To our health research partners across the country, our Board of Judges, Dr. Erna Arrollado of the National Institutes of Health, University of the Philippines, Manila, Dr. Myra Villarreal of the Bicol University, and our very own Dr. Albert Remus Rosana from the Department of Science and Technology, Philippine Council for Health Research and Development, to our moderator, Dr. Giancarlo Malabad, our participants, magandang hapon po sa inyong lahat. As one of the implementing agencies of the Philippine National Health Research System, it is always a pleasure for us to witness the growing potential of our young Filipino researchers through the oral research paper competition held every PNHRS week. The competition supports the system's vision of creating an enabling environment for health research by providing a platform to nurture the talents of researchers and showcase their works early in their career. Today, we specifically highlight studies supported by the Council's Undergraduate Thesis grant in natural products. Through this program, we aim to train young individuals in the conduct of research on natural products and eventually build a pool of well-rounded researchers in the field. Along with assisting our young researchers in the conduct of research, we believe it is equally important to allow them to gain experience in communicating their studies, which is the focus of today's activity. On behalf of the DOST, Philippine Council for Health Research and Development, congratulations to all of the participants for this year's competition. Undertaking health research is a significant feat by itself, and being here to present your work to our esteemed panel of judges is another achievement worth celebrating. I hope that this experience will further advance your interest and passion for research and soon lead you to careers in health research. I am also confident that you will also serve as an inspiration to your peers, encouraging them to explore the challenging but fulfilling world of research. We look forward to seeing you being part of our community in the future and joining us in the mission of making Filipino lives better. Throughout the years, our researchers have proven that indeed our talents across the regions are more supporting. In turn, the Council will always support Filipino researchers at all stages in realizing their potentials and building their capacities in health research through the programs such as the Undergraduate Thesis Grant and the Oral Research Paper Competition. This 15th PNHRS Week, we will showcase more activities in the health research community, highlighting the theme, Health Research Responding to the Challenges Towards National Recovery and Resiliency. We hope that you can also tune in to the upcoming sessions to learn more about the current updates on Philippine health research. I wish you all a meaningful week ahead. Maraming salamat po at mabuhay ang Philippine National Health Research System.
Thank you very much, Dr. Montoya, for that warm welcome. Once again, we welcome all our audience, those who are in the Zoom, and also those viewing us via Facebook Live. Ayan, nakakatuwa po, no? We are reading from your posts in Facebook Live through our DOST PCHRD Facebook page. Ayan, so people are cheering, go Region 9, go Region 5, go Region 10. Ayan, so keep your cheers coming and let your regional representatives know that you are supporting them in this afternoon's competition. All right, so i-cheer po natin sila and let them so that their confidence will be boosted. All right, now before we start the competition, let me present the contest guidelines, the criteria for judging, the mechanics, and of course, the prizes for our winners. All right? Okay, so all the thesis projects funded under the 2019 and 2021 run of the DOST PCHRD Undergraduate Thesis Grant in Natural Products were included in the shortlisting process. But only the top 6 grantees with the highest average scores from the technical evaluators were selected to present in this afternoon's competition. So these are the criteria for judging. We have 25% for the mastery of subject matter, 20% for the content of presentation, 15% for the manner of presentation, 10% for the organization of presentation, 20% for the ability to answer questions later in our Q&A portion, and another 10% for the use of time. And those total to 100%. All right? Now let's proceed to the other mechanics. The submitted slide deck to the PCHRD Secretariat is already final and will be presented this afternoon. Slides must be limited to a maximum of 20 slides only and must not have any slide transitions, animations, or movement of any description. There should be no additional electronic media such as sound and video files. All right? And the principal investigator must be the presenter during the contest proper right this afternoon. Moreover, presentations are limited to a maximum of 15 minutes only. So be very mindful of your time, presenters, okay? Competitors exceeding 15 minutes will have deductions on their final scores. And that would be 1 point deduction for every. So take note of that. Presentations are considered to have comments when a presenter starts their presentation through speech, okay? And during the presentation, the finalists shall open their cameras and shall maintain eye contact with the panel of judges and the audience by looking into the camera while speaking. Okay? Now, there will be a 10-minute Q&A session after each presentation where our board of judges will be throwing their questions to our competitors. After all the presentations, an unbanked discussion with the board of judges will be conducted to determine the top 3 winners. Okay, so we will have top 3 winners who will be awarded for this competition. The decision of the board of judges is deemed final. All right. Now, the most exciting information that our competitors need to know. Okay, for our prizes, the top one or the first place will receive a cash amounting to 30,000 pesos, and 20,000 of that will be for the student, and the remaining 10,000 pesos will be for his or her advisor. All right, so not just the student is the one winning, but also the advisor. All right, now for the second placer, he or she will receive 22,000 pesos, 15,000 pesos of which will be for the student, while the 7,000 pesos remaining will be for the advisor. The third placer will be receiving 15,000 pesos, 10,000 of which will be for the student, and the remaining 5,000 will be for the advisor. All right, but to the other 3 finalists who will not get any place, don't be sad because you will still receive 5,000 pesos each. Okay. Now, the top 3 finalists or winners will be awarded with a certificate of recognition, and the other finalists who won't make it to the top 3 will be provided with a certificate of participation. All right, so I hope that is clear and that somehow motivates our competitors and finalists for this afternoon to do your best so that you can make it to our top 3. All right? All right, so now that I have presented our contest guidelines, the criteria for judging, and our prizes, and as well as the mechanics, I will now proceed with the introduction of our esteemed board of judges. All right, now let's begin with Dr. Erna C. Arrollado. Okay, Dr. Erna Arrollado is a professor at the College of Pharmacy of the University of the Philippines, Manila, and also the director of the Institute of Pharmaceutical Sciences at the UP Manila National Institutes of Health. Her researches are focused more on drug development from natural sources, which led to several intellectual patents as utility models. One of which is the hemoxeter, which is being used by patients who have blood problems, particularly those who have very low hemoglobin levels, like those with anemia, leukemia, alpha thalassemia, and those who are undergoing dialysis. As a prolific researcher, she was recognized as an outstanding researcher by different institutions such as the National Research Council of the Philippines, the Philippine Pharmacists Association, the University of the Philippines System as University Scientist, the Gawad Chancellor Award for Research by UP Manila, Outstanding Alumni in Research by the College of Pharmacy UP Manila, and the University of San Agustin, Iloilo City. In a few days, she will be recognized by the DOST-PCHRD as the best mentor in health research in the NCR cluster and the national level. So advance congratulations to Dr. Arrollado. Welcome, ma'am, to the competition. Thank you very much for joining us in this afternoon's event.
Thank you.
All right. Salamat po.
Thank you, Dr. Malabad.
Opo. Thank you, ma'am. Now, next in our list of the Board of Judges, we have Dr. Albert Remus R. Rosana. Dr. Rosana is currently the Science and Technology Fellow for the Tuklas Lunas Drug Discovery and Development Program at DOST-PCHRD. He also currently holds or concurrently holds a university professorial lecturer and adjunct full professor at the University of the Philippines, Los Baños and Open University. Dr. Rosana secured his doctoral degree in chemistry in the field of microbial chemistry and biotechnology from the University of Alberta, Canada. He is a Killam Laureate awardee, a Sir Alexander Graham Bell, and a Major General George Vanier Scholar in Canada. His research was focused on the interdisciplinary interface of microbial genomics, transcriptomics, and metabolomics, forest entomology, fungal synthetic biology, and natural product chemistry. He also obtained his Master's of Science in Microbiology and Biotechnology at the University of Alberta, while his bachelor's degree in biology at the UP Los Baños, graduating magna cum laude. He was recently awarded the 2021 Gawad ng Pangulo, Pamana ng Pilipino, the most inclusive and highest honor given by the Philippine government to Filipinos based overseas, recognizing his expertise in the field of biology and biotechnology, as well as his ingenious research in fighting the mountain pine beetle epidemic, which is an invaluable contribution to preserving Canada's ecosystem and Earth's battle against climate change. Let us all welcome Dr. Albert Rosana. Sir, good afternoon po, and thank you for joining our competition as one of our judges.
Thank you very much, Dr. Malabad. Yeah.
Ayan. Thank you, sir. Thank you po. Now, last but definitely not the least, in our from our board of judges, we have Dr. Myra O. Villarreal. Dr. Villarreal obtained her BS Agriculture and MS Microbiology degrees from the University of the Philippines, Los Baños, and her PhD in Agriculture from the University of Tsukuba, Japan. She is currently a Balik Scientist of DOST-PCHRD and affiliated with the Faculty of Life and Environmental Sciences of the University of Tsukuba, Japan. Dr. Villarreal is engaged in functional food research, especially in the use of natural products from foods for health and has authored and co-authored more than 30 publications in the field. Let us all welcome Dr. Myra Villarreal. Ma'am, good afternoon po, and thank you for joining our competition this afternoon as part of our Board of Judges.
Thank you for having me. Good afternoon, everyone. Good luck to all the presenters.
All right, maraming salamat po, Ma'am Myra. Now that I have presented our contest guidelines down to our Board of Judges, let us now proceed to the contest proper. Ayan, nako, kinakabahan na siguro ang ating mga finalists ngayong hapon na ito. But don't worry, you have been selected as the top 6 of all our undergraduate grantees. So you've done a good job already having been part of this year's competition. All right, so let's begin. And our first finalist is Jason C. Alcano. Jason graduated with a bachelor's degree in chemistry from the Central Mindanao University, Musuan, Maramag, Bukidnon. Ayan, last June 2022. For his undergraduate thesis, he did the first metabolomic profiling of 2 understudied edible ginger plants, locally grown at Lingera, Eliachor, and Philippine endemic Cornstedia conoidea, under the mentorship of Dr. Gina Barbosa. His research interest expands from natural products chemistry, metabolomics, to analytical chemistry. Presently, Jason is working as a science research specialist at the Research and Development Center for Maternal and Child Health, Davao Medical School Foundation, Incorporated in Davao City. Jason will now present his completed study entitled Exploring the Philippine Endemic Hornstedia cornoidea fruit and Etlingera elator inflorescence, its metabolite profile and antioxidant activity, and its potential for functional powder development. So Jason has opened his camera and I think he's ready to present his paper, right, Jason?
Yes, sir.
All right, so let's all give Jason a warm round of applause as he begins his presentation of his study. Go ahead, Jason. The floor is now yours.
Mic test. To the esteemed members of the panel, to my co-researchers, and to the virtual audience, ladies and gentlemen, good afternoon to you all. As I read this year's PNHRS Week theme, which is Health Research Responding to Challenges Towards National Recovery and Resiliency, it's only fitting to tap our own biodiversity, our local plants, to look for to respond to our health and food security by looking at alternative for medicine and food sources. Nature, through plants, provide food and serve as a haven for medicinal compounds. Hi everyone, my name is Jason Alcano. I'm a representative from the Central Mindanao University in Region 10. And I will be presenting to you my undergraduate thesis entitled Exploring the Philippine Endemic Hornstedia conoidea fruit and Etlingera elator inflorescence, its metabolite profile, antioxidant activity, and its potential for functional powder development. So here is the outline of my presentation. For the introduction, free radicals could induce increased oxidative stress, DNA damage, protein carbonylation, lipid peroxidation, which is linked to manifestation of aging and inflammation and of serious diseases like cancer, cardiovascular diseases, atherosclerosis, diabetes, neurodegenerative diseases, and chronic fatigue syndrome. While normal levels of free radicals in our body have a biochemical importance, free radicals can be derived from exposure to environmental factors like ionizing and UV radiation, through food, through diet, and even through air and water pollution. Next is that how do we go about this problem? So we use supplementary antioxidants. So in this study, we ventured to nature to look for natural antioxidants. In this study, we utilized the family of Zinnibraceae, in which Hornstedia conoidea and Etlingera elator belong to. Hornstedia conoidea is also known as panoon, litokon, or tagbak in our local language, and Etlingera elator inflorescence is also known as the common torch ginger plant or the Philippine wax flower. We use metabolomics to study these plants. So metabolomics is an indispensable tool in natural products research. It has been used for quite a while already. And this study, we use metabolomics to investigate the bioactive compounds that can be derived from these plants and for the potential development of a functional powder from these plants. For the objectives of this study, the main objective of this study is to do the first metabolite profiling and examine the total phenolic content and total antioxidant activity of Ethninia elazur inflorescens and Hornstetia conoidea fruit. Also, specific objectives include develop functional powder, perform untargeted metabolite profiling using GCMS and LCMS-MS, analyze data using multivariate and chemometric techniques, determine the TPC and TAC of the plant studied, and compare and correlate the TPC and TAC of the plants studied. For the significance of this study, the study is towards bioprospecting for drug leads. It has an indigenous relevance because Hornstetia conoidea is also consumed by the Tigaonon tribe of the Agusan del Sur of Bukidnon. It can give ethnomedicinal insights towards the ethnobotanical use of the plants. It also generates new scientific body of knowledge. So it's going to fill in the research gaps in literature. And lastly, it has the potential to provide product income generation for product development to our local and tribal communities. So just a short summary of the methodology. From sample collection, sample were drenched with liquid nitrogen. For the sample preparation, it was done following the method of Barbosa et al. 2017, which is adapted from the protocol of the University of Melbourne and the metabolomics platform in University of Melbourne, which includes homogenization of plant tissues under liquid nitrogen condition, sequential extraction with methanol and water, then centrifugation, phase separation, and speed vacuum for solvent desolvation. The next is the GCMS and the LCMS-MS metabolite profiling, which was done using Agilent equipments. And then GNPS was used to putatively identify the compounds that can be derived from these plants. And then chemometric analysis was done using MetaboAnalyst 5.0. For powder development, commercial food dehydrator was used with dehydration parameters that were cited in the literature. For spectrophotometric assays, Paulin-Chocolate assay and phospholipidem assay was used for the spectrophotometric assays of the plant extracts. And for the statistical data analysis, the data were reported as mean plus HT. T-test, Grubbs test, and Pearson R correlation was also used for the purpose of the statistical analysis. For the results and discussion, so here is the sample description and the product development. So as you can see here in the images, in the 4 images here from A to D, we have from A to C, that's the fresh and the dehydrated powder from Hornstetia conoidea, and from B and D, that's the fresh and dehydrated powder from Ethninia elazur. So as you can see here in the right side of the slide, we made a product prototype for this purpose of the presentation. For the gas chromatography mass spectrometry, metabolite profiling was done, and the identification was based on the NIST metabolite match library matching, which is based on the reverse match and match score of greater than 700, and also manual curation of the spectra against the database spectra. So table 1 shows the GCMS and NIST identified metabolites for E. elazur inflorescens, in which notable compounds are lauryl alcohol, which exhibits in literature antimicrobial properties, and beta-cytosterol, which exhibits anti-diabetic activity in silico, in vitro, and in vivo studies using streptozotocin-induced diabetic rats. For table 2 shows the GCMS NIST identified metabolites from H. conoidea fruit. We have furfural and 5-hydroxymethyl furfural, which are the notable compounds we can found here, which shows antioxidant, anti-tyrosinase activity, and a lot more activities. Also, oleic acid and gamma cytosterol was also detected for H. conoidea. For the liquid chromatography mass spectrometry, which was done using ultra-high performance liquid chromatography quadruple time of flight mass spectrometry, that table 3 shows the metabolite profiles that were putatively identified. So the chemical structures are presented here. So some of the notable compounds are cyanidin-3 glucoside, which exhibits a wide array of bioactivities like It inhibits cyclooxygenase 2 inhibition expression in raw 264.7 cells in literature. In literature as well, it ameliorates type 2 diabetes through this induction of differentiation of free adipocytes into smaller and insulin-sensitive adipocytes. We also have chlorogenic acid, which exhibits a wide array of biological activities. It has antioxidant activity. It's a hepatoprotective, cardioprotective, anti-inflammatory, antipyretic, neuroprotective, anti-obesity, antiviral, antimicrobial, and among other bioactivities, which is reported in literature. The cumulative biological activities of the identified compounds are reflected in the slide. For the LCMS-MS metabolite profiling for the H. conidaea fruit, the same workflow was also done. The following in the table 4 were the putative identified compounds from H. conidaea fruit. Presented here are the chemical structures of the putative identified compounds, which include notable compounds with great biological activities like procyanidin A2, which exerts anti-inflammatory and antioxidant activity in liposaccharide-stimulated raw 246.7 cells in literature. Also, we have quercetin 3-orotinoside here, which is rutin, which is commonly found in plants. It has an antimicrobial. It is a strong radical scavenger. It's also a hepatoprotective agent via promoting antioxidative and anti-inflammatory properties against oxidative liver damage, which is reported still in literature. So we also did the chemometric analysis using the principal component analysis. As you can see here in the visuals, PCA score plots of Epinera elazor inflorescence in Poinsettia conidea fruit in positive and negative mode shows no overlap, and there's complete separation, indicating the uniqueness of the different profiles overall. For the chemometric analysis as well as through clustering analysis and heat maps, it identified was done, and it identified markers that are high in both markers that are high in fresh and dried samples for both plant samples. For example, in Epinera elazor, we have the metabolic feature 264.1475 eluting in the indicated retention time, which is higher in dried compared to fresh. The individual box whisker plot here shows the relative levels of specific metabolite in fresh versus dehydrated samples. But as part of the limitation of this study, it needs more time for the identification of this discriminant markers. So this is part of the recommendations already. So shown here is also the total phenolic content and the total antioxidant capacity results, where in the TPC, dehydrated samples were found out to have superior values. This may be due to the increase in dehydrated samples due to the release of bound phenolics and the hydrolysis of complex phenolics, yielding low molecular compounds that have antioxidant activity or that are phenolics. Also, total antioxidant capacity was found out to be higher in the dehydrated samples compared to fresh, and this might be due to the presence of other antioxidant compounds that cannot be detected by the assay. The formation of new components in drying process, like Maillard products, the activation of oxidative and hydrolytic enzymes. So that might be the reason why. Also in this study, we investigated the linear association of the total phenolic content and the total antioxidant capacity values of all the data set that we gathered. So it has been found out that there's a strong positive association of TPC and TAC values with an R squared of 0.8953 and a correlation coefficient R of 0.94.62, which is close to unity. For the conclusion and recommendation of this study, this is the first and pioneering study to report the GCMS and LCMS-MS metabolite profile of the plants studied. Putatively identified compounds of H, C, and EE are reported to have wide array of biological activities in literature. And for the purpose of the spectrophotometric assays, dehydrated plant samples were found to have superior values compared to its fresh counterparts. We are in the better position to say that dehydration may have been proven to be effective in the preservation of metabolites and elicited better activity. So the study also further explored the development of products from locally sourced edible plants. As part of the recommendation, one of the recommendations is to identify the detected discriminant molecular features from the chemometric data to better understand the effect of the dehydration process, and to evaluate as well the part of the recommendation is to evaluate the proximate and elemental analysis, sensor profile analysis, and the potential of the product developed. And for subject the sample extracts to more reliable assay in vivo and silico to prove the functional activity of it. And last, do market research and feasibility studies for the purpose of commercialization in the future, in the way future. Before I end this presentation, I would just like to make some acknowledgement to the DOST PCHRD, DOST SEI, to the PMS of mass spectrometry, to the Center, to the Natural Products Research and Development Center of CMU, to CMU TL Tuklas Lunas and Development Center Project 5 for the use of equipment, to the Northern Mindanao Consortium for Health Research and Development for the support, and for everyone that took part to this to the success of this study. I thank you all. And for this, I end my presentation. Thank you for listening to this presentation. That would be all. Thank you.
All right, dagang salamat, Jason. Region 10 must be very proud of your accomplishments and presentation this afternoon. Someone in the Facebook Live said, Region 10 is rooting for you. All right, and you still have a few seconds left in your presentation. But good job. Now let us proceed with the question and answer for Jason. And for this round, we will have two of our judges to throw questions to Jason, for Jason to answer. All right, we'll have Dr. Arrollado to be followed by Dr. Rosana. Dr. Arrollado, can we have your questions first, ma'am?
Okay, so my first question is, what is novel in your study as compared to the previous studies done on gingiber family?
For this, I think, first of all, thank you for that question, ma'am. For this study po, we used the understudied and locally grown Zingiberaceae plants. For the purpose of novelty, that's it. The novelty is the use of understudied plants. But for the purpose of the compounds that were identified, though all compounds were found to be known and to have a wide array of biological activities, it is part of the limitation of the study that we didn't really had. It's not really part of the study to have to identify novel compounds from these plants. Yes, po.
Okay, so shall I follow another question, or it will
Yes,
be an
ma'am.
alternate asking?
You can
Okay,
ask another question.
Okay, so another question is, what is the purpose of profiling untargeted metabolite? And first, what is untargeted metabolite?
Okay po. Again, thank you for that question, Dr. Arellado. For the benefit of everyone, when we say untargeted metabolite profiling, we're not identifying some of the compound classes that we are to identify. So we can just inject the sample to our LCMS and then look at all the metabolites that can be identified from the profile. So the purpose of metabolite profiling is to just skim through the rough picture of what are the metabolites that can be found to this plant's extracts. And further, if we can find some interesting metabolites that cannot be ID, or that can be ID by the targeted metabolite profiling, we can go on with the next step, which is the targeted metabolite profiling. If we have compounds that we want to target and then identify and quantify. I guess that's it, ma'am.
Okay, last question. How do you use chemometric techniques?
For the chemometric techniques po, since we're dealing, metabolomics entails with large data set. For the purpose of more, how do we say this, a more systematic approach to big data, we use chemometric analysis to identify the discriminant markers, the interesting metabolites, the differentiating metabolites, which we can focus later on identification in the same way in this study as well. So chemometrics data analysis would give you a much systematic approach and a more clearer view of what you're looking for.
Okay, thank you, Jason.
All right, thank you,
Thank you po.
Jason, and thank you, Dr. Arrollado. Now, can we have Dr. Albert Rosana to throw his questions for Jason?
Hello, good afternoon. Can you hear me?
Yes, yes.
Yes po.
Yeah,
Good afternoon po.
Okay, first and foremost, congratulations, Jason. That's actually a journey to combine both the Plus Luna's sort of drug track as well as the functional food, because you managed to develop actually products based on this inflorescence and fruit of the two Singapuraceae family. I guess along that line, my first question is, how will you be able to establish the safety of this functional food, the powders that you have? Maybe you can provide an avenue or idea how you can maybe ensure the safety of these products.
Yes, po, sure po. Thank you. First of all, thank you for that question, Sir Dr. Rosana. Since the functional powder is just a prototype, the commercialization is way ahead, way in the future back. So we really need to also assess the safety of these kinds of products from the plants. Though ethnobotanical knowledge would suggest that these are edible plants, because our tribal communities and local communities are already consuming them. But we just have to make some scientific validation as to their safety. So maybe we can do a lot of cytotoxicity assays to normal cell lines, and even do admit, if possible, how does the bioactive compounds, or even in these plant samples, are being processed in the human body. And also, one of the things that we also have to look at is the inorganic components that can be found in these plants, because they are Zingiberaceae plants. They are directly above the ground. So maybe they have bioabsorption capacities or anything like that. So we have to also measure the heavy metals and assess if there are beyond the levels that are required by the FDA or any other governing bodies. So I guess that would be an idea to start with.
That is quite clear. You seem to know the protocol for drug discovery and development. Thank you for that, Jason. My second question is on the utilization of the MetaboAnalyst platform that you have. The depth by which you can pick certain metabolites in your untargeted approach may have certain limitations. Can you please comment on not just the establishment of the signals on your LCMS, but also the database component for this MetaboAnalyst platform?
First of all, the MetaboAnalyst platform is used for the XMS spectral analysis. So there's a lot of things that you have to consider when it comes with using MetaboAnalyst. From what I have learned throughout my journey, one of the things is that you have to manually curate your data first, especially when you have to blank subtract first, and then for identification of the compounds, I also connect it with the GNPS platform, the MetaboAnalyst and the GNPS platform. While it is true that GNPS has been utilized by a lot of scientists in the natural products chemistry field and in other fields as well, it still has the limitation because maybe in the databases the collision energy is different, so it's going to be the fragmentation pattern of the compounds will be different as well, and also for the identification of novel compounds are not in the database. That's still a limitation as well. So for the purpose of a gold standard validation of the identification of compound, we can go on with the isolation process and do an NMR study and experiment to do structure elucidation for that purpose.
Thank you very much. Those are my only questions.
Thank you, sir. Thank you for that question, sir.
All right, maraming salamat, Dr. Arrollado and Dr. Rosana. And also, Jason, thank you and a good job to your presentation and how you answered our judges' questions. All right, so maraming
Thank you
salamat
po.
and congratulations again. Let's give Jason a virtual round of applause. Ayan, makaka-relax ka na, Jason, as we as you witness the next 5 presenters ngayong hapon. All right, so
Thank you po.
that's the beauty of being first to present. Okay, all right. So without further ado, let us start with our second presenter for this afternoon's competition. Our next finalist is Mr. Gerson Angelo Armesin. He is a graduate of Bachelor of Science, major in Biology from the Velez College in Cebu. Since his freshman year, he had developed the love to serve the Velezian community and the rest of the society using the information he had learned from his institution and the scientific breakthroughs shared within the scientific community. Being raised in a family with great appreciation and love for agriculture, he takes pride in his and his colleagues' research project about the antimicrobial properties of Carabao mango Mangifera indica. After attaining his bachelor's degree, Mr. Armesin is now pursuing his dream of becoming a physician with his admission to the College of Medicine within his locality of Mandaue, Cebu. Mandaue City, I should say. He will now present his completed study entitled In Vivo Assessment of Antimicrobial Effect of Mango, Mangifera indica pills, and seed extracts, and Lactobacillus delbrueckii on Streptococcus pyogenes and Shigella boydii inoculated in albino mice, Mus musculus. Gerson, I think you're now ready to present, right? Okay, make your region proud. Let us all welcome Mr. Gerson Armesin as he presents his study. Go ahead, the floor is now yours, Gerson.
Bacterial infection and their treatments are gaining medical scientific concern due to the prevalent antibiotic resistance of many bacterial species owing to bacterial ability to develop resistance to antimicrobial agents through excessive, frequent, and unnecessary exposure. Good afternoon, everyone, especially to the PCHRD and to the board of panelists and to our MC. I will be presenting the study in vivo and in vitro assessment of antimicrobial effect of Mangifera indica L seed and peel extracts and Lactobacillus delbrueckii strain on Streptococcus pyogenes and Shigella boydii inoculated in Mus musculus or albino mice. Due to the increasing trend of antibiotic resistance, it had now led to the exploration of plants as antibiotic agents. Streptococcus pyogenes causes a diverse range of clinical manifestation, and it showed that it is resistant to treatment that may lead to the infection to severe cases and recurring infection. Additionally, according to Chin 2000, 600,000 deaths worldwide are caused by Shigella species such as Shigella boydii. Hence, acquiring antimicrobial effect of Mangifera indica and Lactobacillus delbrueckii will be one step closer to catching up with the increasing trend of antimicrobial drug resistance. This scientific investigation has one main objective, which is to assess the antimicrobial effect of Mangifera indica L seed and peel extracts and Lactobacillus delbrueckii strain against Streptococcus pyogenes and Shigella boydii in vitro and in vivo. Additionally, it has 3 sub-objectives presented, which includes measuring the mean zone of inhibition and comparing the significant difference of all the test treatments against both S. pyogenes and S. boydii, and also to examine the antimicrobial effect of the treatment with the highest zone of inhibition against both S. pyogenes and S. boydii in vivo, and comparing the means across the treatments. Now we go to the significance. This study would benefit these 5 institutional areas. The academe, as the results of this study, would serve as reference information on related to antimicrobial studies. The medical field, as the results may provide potential alternative on inhibitory agents against S. pyogenes and S. boydii, as well as give light on health-oriented potential of L. delbrueckii found common on dairy and yogurt products. Future researchers may also benefit in these results, as it may serve as groundwork for future studies on antimicrobial activity. The environment, because the results of this research may generate new means of utilization of mango waste aside from conventional disposal methods. Lastly, the government, because this study may generally encourage policymakers to implement projects to sustainably utilize mango seeds and peels as antimicrobial agents and as a source of antioxidants to generate new drugs. To achieve the objective of this study, these methodologies were utilized. Mango seeds and peels were collected from ProFoods Mandaue City, Cebu, and ethanolic extraction was conducted at the Valles College Biology Laboratory. And experimental testing was also carried out at the University of San Carlos, Talamban. Microorganisms S. pyogenes and S. boydii were procured from Phil Anaserv Incorporated, while isolates of L. delbrueckii was procured from USC Microbiology Laboratory. The albino mice was also procured from the USC Animal House Talamban campus. Prior to processing for Kirby-Bauer susceptibility assay, 4 treatments were prepared using ethanolic extraction, namely the mango seed and peel extract, mango seed and L. delbrueckii treatment, mango peel and L. delbrueckii treatment, mango seed and peel extract, and L. delbrueckii treatment alone. The Kirby-Bauer susceptibility assay was utilized in vitro assessment of antimicrobial effect of M. indica and L. delbrueckii. It utilized 14 blood agar plates and 7 of which streaked with S. pyogenes and 7 streaked with S. boydii. For each in vitro inoculum, 9 discs of treatment of positive, which is ampicillin, distilled water for negative, seed extract treatment, peel extract treatment, seed and peel extract treatment, L. delbrueckii alone, seed extract and L. delbrueckii, peel extract and L. delbrueckii treatment, and seed and peel extract and L. delbrueckii treatment were placed equidistantly. For the in vivo mice segment, the researchers patterned its procedure from Venkatasan et al. 2005, and the volume and concentration of inoculum introduced was based on Choi et al. 2011. In the in vivo segment, 48 mice, 36 males and 12 females, were divided into 4 groups per inoculated pathogen, S. pyogenes and S. boydii. The 4 groups included the experimental, treated with conventional the experimental treated with mango and seed peel extract and L. delbrueckii, positive, treated with the conventional ampicillin, and the negative, treated with normal saline solution, and lastly, the untreated group. The pathogens and the treatments were inoculated via oral gavage needle. Terminal cardiac puncture required aspirating blood samples from each anesthetized mice. The obtained samples were then processed for WBC differential count at the Cebu Veterinary Doctors Ramos branch. Lastly, the experimental data gathered both in vitro and in vivo were treated using one-way analysis of variance. Post hoc Tukey's comparison test was also employed, and along with the one-way ANOVA, it was carried out using SPSS version 25. Pairwise comparison t-test was also used via GraphPad software. Now we proceed to the results. Here we are presented the Kirby-Bauer susceptibility results. Percentage shows that the S. pyogenes and S. boydii, the positive treatment ampicillin, or what you can see in the table as AMP, had the highest mean zone of inhibition at 40 mm and 31.5 millimeters. Additionally, the seed and peel extract combined with L. delbrueckii, or SPLD, resulted in the largest zone of inhibition for both S. pyogenes and S. boydii among experimental treatments. The table presents that when compared to the negative treatment, the in vitro assay, which was distilled water, all treatments except L. delbrueckii had a significant or differing value. Additionally, the table also suggests that relatively low antimicrobial activity depicted by the treatments are still sufficient to be significantly different from the complete non-activity demonstrated by the negative treatment, which was distilled water. Therefore, what we can draw out from here is that L. delbrueckii alone had non-existing antimicrobial activity against both S. pyogenes and S. boydii. This table presents the mean difference of comparison t-test for both S. pyogenes and S. boydii. Of all the treatments utilized, ampicillin showed statistically significant difference across the 2 pathogens. It had a value of 40 for S. pyogenes and 31.5 for S. boydii, implying that ampicillin is indeed a more potent inhibitory agent for S. pyogenes than S. boydii. Here represented on the left the tabulated zone of inhibition per treatment for S. boydii, and an important information here is that ampicillin, AMP, had the highest mean zone of inhibition, and the most important information is that it is followed by SPLD, and that L-delbruckii or LD treatment alone had lowest mean zone of inhibition. Here we are also presented on the left the tabulated zone of inhibition per treatment for S. pyogenes, and important information here is that similar to S. boydii, ampicillin had the highest zone of inhibition, most importantly followed by SPLD, and that L-delbruckii treatment alone had the lowest mean zone of inhibition. The in vivo assays were divided into 4 groups per inoculated pathogen, namely the experimental, positive, negative, and untreated. For the body scoring, it was merely subdivided into attitude, or the way that the mice acted throughout the observation period, and weight, the increase or decrease of mass thereof during the observation period. For the attitude, a score of 1 would mean that a mice is lethargic or does not move, while 5 would mean that it was overactive or erratic. For the weight, a score of 1 would mean that the mice had muscle wasting, and a 5 would mean that the mice were obese. In the test groups inoculated with S. pyogenes, all mice were active and normally behaving all throughout the observation period. For the weight, the negative group had the highest mean at 27.12 grams. For the group inoculated with S. boydii, the mice in each test group acted and behaved also normally throughout the observation period. And for the weight, still, the negative group had the highest mean at 25.43 grams. Here we are presented the summary means, standard deviation, and analysis of variance of the differential WBC of test mice inoculated with S. pyogenes. It is important to note that the mice treated with SPLD had marked effects on the WBC levels along the platelet or PLT count recovered during the cardiac puncture. This significantly is comparable to the negative and untreated groups. Additionally, here we are presented the summary of means and analysis of variance inoculated with S. boydii. It is apparent that the mean of WBC concentration collected from the mice received SPLD inoculated with S. boydii have a considerable margin compared to the negative and untreated groups. And this is simply a comparison of the post-hoc analysis and by Tukey's honesty significant difference. For the mice inoculated with S. pyogenes and S. boydii, the positive treatment shows a statistically significant difference relative to the group who received negative treatment or NSS. Importantly, the group that received experimental treatment or SPLD showed no statistically significant difference relative to the mice group that received the positive treatment or ampicillin. This suggests that antimicrobial activity of ampicillin is associated to SPLD in inhibiting the growth of both S. pyogenes and S. boydii. Here we are simply presented the antimicrobial activity of SPLD and all the rest of the treatments compared to the untreated group. Generally for this study, the researchers took inspiration in 2 main situations. First, that there is an emergent concern for antimicrobial drug resistance and that there is a need to combat it, and that the Philippines, as a mango exporting country, produces significant amount of organic waste that may be utilized to combat the aforementioned problem. In this study, it was found in a Kirby-Bauer assay that the most effective extract against S. pyogenes and S. boydii was the combination of M. indica seed and peel extract with L-delbruckii or SPLD. Now, based on the study's result, here are our conclusions. It was concluded that the combination of M. indica seed and peel extracts with L-delbruckii have a statistically significant antimicrobial effect against S. pyogenes and S. boydii. Additionally, L-delbruckii alone had non-existing antimicrobial effect against S. pyogenes and S. boydii. Lastly, it was found that SPLD showed no statistically significant difference relative to the mice group that received the positive treatment or ampicillin, and that there is a significant increase of the WBC count that can be attributed to the tendency of SPLD to have immunostimulatory effect on host immune response, consequently aiding in the fight against infection. For a recommendation, the researchers would recommend that the future researchers utilize long observation period both in vitro and in vivo to gain more elaborate results. Also, banking on the increase on differential WBC count, the researchers would suggest to explore the potential immunomodulatory activity of M. indica. The researchers also suggest to include toxicity assays to test for minimum lethality of M. indica to live organisms, and also the use of different species for lactobacillus. We also encourage future researchers to utilize other means of extraction method for M. indica plant parts. The next time you see a mango on your countertop, I hope that would change your thinking that it's not just a fruit, it may be a superfruit. This will be all for the research presentation. Thank you once again, and good afternoon to all of us.
All right, dagang salamat, Gerson, and thank you for your and good job on your presentation. All right, now let's proceed immediately to our question and answer portion for this round of our competition. We will also have two judges who will be asking their questions for Gerson, and we will have Dr. Villarreal and Dr. Arrollado. Okay, can we have the questions first from Dr. Villarreal? Ma'am Myra, go ahead po with your questions.
Okay. Congratulations, Gerson Angelo, on your presentation and for the completion of your study. And of
Thank you,
course,
Doc.
being granted the funding to do your research is already an achievement. So again, congratulations. I have several questions. I don't know if I should ask them all, but I think the first thing is mango is not unique to us in the Philippines. We share it with India, Thailand, and other Asian countries. So my question is, so what makes your study unique? Because there are also other reports on the use of peel and other parts of mango as far as antimicrobial effect, and other studies have been done on other things as well. So what makes your study unique in this aspect?
Well, what I've learned throughout the entire journey in this investigation is that the previous studies on mango really focus on the bark, especially on the leaves, most that are common on the outside. While what makes our study even more unique is that it combines Aspergillus and Aspergilli as the pathogens of concern, which is also common in our country, such as fever and dysentery. So I think the focus and the combination of both, especially with the introduction of Lactobacillus delbrueckii, which is commonly found in yogurt products, is what makes our study unique for me and based on what I have learned throughout the journey.
Okay, thank you. Is it okay to ask more questions?
Yes.
Okay, so the other one is you use these two microorganisms, and how long did your study last? I mean, in terms of months, how long did you perform your experiments using these two microorganisms?
Also, these microorganisms were already procured from Phil Anasir, ma'am, and
yes. So how long did you use the cultures?
The cultures were only available for 6 days, ma'am, in total.
You used them for 6 days only, or did you use them for several weeks or something?
6 days only, ma'am. It was due to the limitation, also the availability of the lab.
Okay, because my concern is, did you at certain point check whether the cultures that you're using are the original ones that you received? So did you check at least as far as?
Yes, ma'am. Aside from receiving the authenticity certificate, we also had the assistance of the laboratory technologists with us in identifying and not just physically and also microscopically, ma'am. Thank you.
Okay. Okay. Last one. Yeah.
Don't worry. You can ask more questions.
Okay. Can I ask more? Okay. So maybe two more. Okay. So another one is, I noticed that you did not do any dose-dependent study. Is that right? You didn't do any dose-dependent assays were not performed.
Yes, ma'am. We only patterned into a particular study.
Yeah, and that's why ampicillin is winning all the time in all your studies. So why is that? Why did you not perform a dose-dependent assay because that would be the most, I mean, probably the first thing that I would recommend to a student if a student is doing something similar to what you've done. So is there any reason why you did not perform a dose-dependent assay?
Well, what I can identify really is the main reason, ma'am, is that the time constraint of which the study had to be conducted, and also the availability of the resources and the laboratory, ma'am. And we were really severely affected with the typhoon, and we really had to revamp and somehow congest and compact the study, ma'am.
Okay, that's understandable. Okay, so maybe just I don't know, you've graduated, but in one of your recommendations, so maybe this is just a comment to whoever will be continuing your study. So you use the absolute ethanol, is that right, as your solvent?
Yes, ma'am.
Okay, so other collaborators would rather work or tamper or play with the concentration of your ethanol rather than change your solvent altogether. So I think that's just what I want to say.
Okay,
so
thank you so much.
To your advisor or to whoever will be continuing your studies, your research is very interesting, and I hope that it gets to be continued. So I think that's all from me. The floor is yours, Dr. Arellano.
Thank you so much, Ma'am Myra Villarreal.
Thank you, Dr. Myra. Okay, so my first question is, why did you use probiotic in your study?
The Lactobacillus delbrueckii, ma'am. Yes.
Yes. Okay. Why did you combine it? Which extract did you combine this? The probiotic. Did you combine it with seed extract or the leaf extract? Please correct me first. There are two extracts that you have prepared, right? From the seed.
There were three main extracts, ma'am. The seed and peel. There were three main
seed
extracts.
And peel.
Yeah,
Okay,
seed
the
and
seed
peel,
and peel.
seed alone.
Okay,
peel
so
alone.
which extract did you combine with probiotic?
So the one that we combined with probiotic, ma'am, was actually almost all prepared because there were 9 in total. So first we combined the seeds and Lactobacillus delbrueckii, the seed extract, then the peel extract with L. delbrueckii, the seed and peel extract with L. delbrueckii, and we also have the probiotic alone. So all the extracts had their combination and were tested with L. delbrueckii against the 2 pathogens, ma'am.
Okay, thank you. Now, why did you use ampicillin as your standard when there are other antibiotics available?
So first, ma'am, it was based on our finding that it was one of the most effective against both the Asparaginus and S. boydii. More so, it was what was available at hand during our research. So aside from the fact that it was one of the most effective during our research, it was also what was really available at our disposal during the conduct of the entire study.
Okay. Did you what is this? Were you interested only on gram positive but not on gram negative?
Yes, so far, ma'am, that was just really the focus on of our scope and limitations, ma'am. So we just focused first on what was commonly found and on gram positive, really, basically.
Okay. Okay. So in your in vivo test, you use 48 mice, right?
Yes, ma'am.
And then you use 36 male and 12 female.
Yes, ma'am.
Right?
Yes.
Why is it that there is no balance in terms of choosing the gender of your test animals?
Okay, so the procurement of those, ma'am, we actually gave it to the animal house already. So the procurement were random, and that was actually based on what was available at 7 weeks, because we have to have them at 7 weeks to be viable in vivo animal models, ma'am. So it was up to the lab technician who provided it to us during the procurement.
Okay. In your procedure, you said that you use 3 milligrams per kilogram of your extract. Is that correct?
Yes, ma'am.
Okay, for your extract. But for ampicillin, you use 7 milligrams per kilogram body weight. How come that you gave more what is this, ampicillin compared with the extract. So you have given more ampicillin when in fact this is already a standard drug. And then for the extract, you use only 3 milligrams. What is the purpose of using that?
Okay, ma'am. So volume-wise, there was more based on what you've asked. So the ampicillin drug that we have was actually in powdered form. So the process by which it has to achieve the same dosage for the extract, we actually still had to convert it or to use distilled water for it, ma'am. Whereas the extracts alone, they were in liquid form, so they were already usable for the inoculation using the oral gavage. So I think that was one of the leading reasons why there seems to be more ampicillin compared to the other extracts that we have utilized in our study, ma'am.
Okay, so we are using those different concentrations for extract and for standard drug. Do you think your result is reliable?
Based on the what I've learned, ma'am, it for me it is because in a way it was only comparing really the efficacy of not only the ampicillin, which is our positive, but also the SPLD or the seed and peel extract to the negative, which is natural saline solution, and of course the untreated, ma'am. So I think in that aspect, for me, it is reliable.
Okay, thank you. That's all.
All right. I was
Thank you
about
so
to
much,
say,
Dr. Ernest.
all
Yes,
right,
ma'am.
maraming salamat. I was about to say that Dr. Arrollado will have her last question, and gladly it's the last question. Ayan, maraming salamat po, Dr. Arrollado and Dr. Villarreal. And of course, good job to you, Gerson, for answering our judges' questions for your presentation. All right, I think Gerson is our only representative from the Visayas, okay, for this year's competition. So good job, Gerson.
Thank you
And
so much, Doc.
you can now relax, okay, as we have our next finalists. All right? So,
Thank you so
maraming
much, Doc.
salamat po
God
ulit,
bless.
Gerson and Ma'am Erna and Ma'am Myra.
Yeah, thank
Now,
you.
let us proceed to our third presenter for this afternoon. We will have a representative from the Calabarzon. All right? He is John Joshua Belen, a graduating BS Biology student from the Southern Luzon State University. His research interests revolve around drug discovery utilizing natural and microbial sources from the Philippines. The initial results of his study were presented and won second best research DigiPoster during the Scimatech Fair 2021, organized by the Philippine Consortium for Science, Mathematics, and Technology. He will now present his completed study entitled Phytochemical Screening and Pharmacological Evaluation of Rhizophora muscronata bark extracts collected from Lucena City, Philippines. All right. So John Joshua has opened this camera and I think you're ready to present, right, Joshua?
Hello, sir. Yes.
All right. So let's all give a virtual round of applause to John Joshua Belen as he presents his study. The floor is now yours, John Joshua.
To the panel of judges and to everyone here today, good afternoon. We are very proud and delighted to present to you the results of our research titled Phytochemical Screening and Pharmacological Evaluation of Rhizophora mucronata bark extract collected from Lucena City, Philippines. Antimicrobial resistance and the insufficient cancer treatments have been two of the most serious public health burdens of the world today. Antimicrobial resistance is a global health threat and remains the greatest concern and biggest challenge to future discovery efforts. Today, the current antibiotic discovery effort is not sufficient enough in delivering new agents at a rate that is sufficient to combat present levels of antibiotic resistance. The same with cancer, which is a multifaceted global health issue that remains second of the most common cause of death worldwide. These two global health issues call for an immediate search for other sources of bioactive compounds that have the ability to to alleviate many serious diseases. Mangroves, they provide crucial roles in the ecosystem, human sustainability, and livelihood. Here in Lucena City, Quezon Province, they are being extensively used by the locals as firewood and fishing stakes. But aside from the ecological and economical uses, mangroves also offer a wide array of biomedical potentials. Mangrove leaves and roots possess rich secondary metabolites that exhibit pharmacological potentials, including antimicrobial, analgesic, anti-inflammatory, anti-cancer, antioxidant, and cytotoxic activities. And upon reading hundreds of journal articles, we have identified research gaps until we have generated this research question. Does the mangrove rhizophora bark also contain phytochemicals and exhibit pharmacological activities? And to answer this research question, here are our objectives. Primarily, we aim to investigate the pharmacological activities of the bark extract and screen its phytochemical constituents. So specifically, we sought to characterize the Rhizophora mucronata bark extract for its phytochemical constituents. Second, to screen the bark extract for its ability to inhibit gram-positive and gram-negative bacteria. Third, to measure the antioxidant potentials of the extract by analyzing its total phenolic content expressed as gallic acid and its radical scavenging activity. Fourth, to assess the toxicity of the bark extract. Fifth, to evaluate the anti-angiogenic activity of the mangrove bark extract in CAM assay through qualitative, semi-quantitative, and quantitative assessments. And sixth and last is to determine the cytotoxic activity of the extract against the AE8 or the Chinese hamster ovarian fibroblast and the A549 human lung adenocarcinoma cell lines. So what are the importance or significance of our study? We believe that this could be a great contribution to the continuous search for other sources of natural-based compounds that can alleviate many serious diseases. It could also be beneficial in further exploring novel bioactive compounds found in mangroves bark. Mangroves grow on public land, however, only a few of them receives any sort of protection. Thus, this research could raise awareness in securing sustainability of our mangrove populations that offer vast array of medicinal potentials. I also believe that it is high time for us to encourage young scholars to delve in the same field of drug discovery research that aim to answer global health issues, because we still have a lot to be identified, to be discovered, and to be explored. We just need more passionate researchers to join with us. And here are our methodologies. We did our collection at Barangay Dalihican, Lucena City, Quezon Province, and authenticated it through the University of the Philippines, Los Baños Museum of Natural History. We then extracted it using ethanol as our extracting solvent. We further extracted it using rotary evaporator to produce the crude extract. After that, we screened it for its phytochemical constituents using manual qualitative test method where color and precipitation reactions were used as an indication. Next is to investigate its pharmacological activities. First is the antibacterial activity using disc diffusion assay, where it was tested against 5 bacterial species: Staphylococcus aureus, Escherichia coli, Enterobacter aerogenes, Bacillus subtilis, and Bacillus coagulans. The total phenolic content and radical scavenging activity was analyzed using the DPPH or 2,2-diphenyl-1 picryl hydrazine assay. For the preliminary toxicity, we conducted the brine shrimp lethality assay, which is based on the killing ability of our extract on the zoological species, which is the Artemia salina. For the anti-angiogenic activity, we conducted the Ductor-Allantoic membrane assay. And for the cytotoxic activity, the MPT assay was implemented. And in answer to all of our objectives, in objective number 1, here is the result of the phytochemical screening. Our study revealed that the bark extract had presence of phytochemical groups, including triterpenes, flavonoids, saponins, glycosides, and tannins. According to Muliani et al. 2019, triterpenes were absent in the methanolic bark extract, but present in our ethanolic extract. So why did this happen? According to Madipur et al. 2014, the yield, biological activity, and even the presence of phytochemical is not only affected by the extraction procedures, but also by the solvent used. Next is the result of the antibacterial activity. Our study revealed that the bark extract exhibited substantial inhibitory action, which was interpreted as very active against Staphylococcus aureus, Escherichia coli, and Enterobacter aerogenes at 100,000 parts per million concentration. However, there was no inhibition recorded against the B coagulans and B subtilis. Moreover, it had the most extensively inhibitory potency among the other mangrove parts, such as the leaves, roots, fruit, and flower. Next is the result of the antioxidant activity. The bark extract yielded 5.32 milligram of gallic acid equivalent per gram of food extract, and the DPPH, or the scavenging activity, exhibited antioxidant activity in a dose-dependent manner. It also had a relatively higher antioxidant capacity than Rhizophora mucronata leaves and roots, with 19.35 IC50, which was interpreted as very strong. The relationship between the DPPH and total phenolic content was evaluated using Pearson correlation test, and the result indicated that there was a very strong positive correlation between the DPPH and TPC. So it only implies that the TPC, or the total phenolic content found in the bark extract, is one of the major contributors of why there was a high scavenging activity. Next, as the result of the toxicological assessment, the bark extract displayed a concentration-dependent toxic activity in brine shrimps and had an LC50 value of 1,500.88, which was interpreted as non-toxic according to the assessment criteria set by Hamidi et al. 2014. Fifth is the anti-angiogenic activity, where we conducted 3 types of assessment: the qualitative, semi-quantitative, and the quantitative assessment. For the qualitative assessment, all the morphological observations were noted. And as you can see in the picture, the DMS order negative control showed tree-like branching pattern, which is an indication of angiogenesis. For the celecoxib, the positive control, it showed thick vessels and no branching patterns, meaning it inhibited the growth of the vessel branches on the poroallantoic membrane. And lastly, the treated with Rhizophora mucronata bark extract indicated abnormal thinning of blood vessels, which is an indication of a moderate anti-angiogenesis. For the semi-quantitative quantitative assessment, independent observers made analysis using a scoring system set by Dara et al. 2018, and it revealed that the bark extract had a weak anti-angiogenic activity at 100,000 parts per million. And to corroborate and justify and support the result of the aforementioned assessments, we did a quantitative assessment where we used AngioTool, an automated quantification framework which further quantifies angiogenesis efficiently. And based on our result, the extract at 100,000 parts per million had promising inhibitory effect comparable to celecoxib with 45.01 and 46.01% inhibition respectively. And it only implies that the Rhizophora mucronata bark extract at high concentrations could be a great inhibitor of progression of blood vessels on the poroallantoic membrane. And the possible reasons by which the extract demonstrated notable anti-angiogenic potentials can be attributed by the presence of phytochemicals and the amount of phenolic content. According to Hassan et al. 2014, tannins and phenols are 2 of the major contributors of high anti-angiogenic activity of many plant and herbal medicines because they act as a complementary agent in the inhibition of blood vessel progression. And lastly, the result of the cytotoxic activity. Our study revealed that the Rhizophora mucronata bark extract had a strong cytotoxic potential against the normal cells AA8, but showed no cytotoxic activity against the human lung adenocarcinoma in high doses. However, it was very interesting that the Rhizophora mucronata bark extract demonstrated moderate but consistent cytotoxic activity in the lowest concentration. It only suggests that the bark extract could be a great inhibitor of cancer cell proliferation even at low concentration. And here is the summary of our results just for everyone to make it more comprehensible to everyone here today. Okay, so since we have 6 objectives, we also have 6 conclusions. We conclude that the bark extract had presence of triterpenes, flavonoids, saponins, glycosides, and tannins. Second, it can be an effective and potent source of antibacterial agents. Third, it could be a good source of antioxidant-based compounds as evident with its very strong scavenging capacity and high correlation value with total phenolic content. Number four, it was non-toxic to brine shrimp Artemia salina. Number five, it had notable anti-angiogenic activity because it had the ability to suppress the formation of new blood vessels. Number six, it had strong cytotoxic activity against the Chinese ovarian hamster fibroblast. However, it showed no cytotoxic effect to A549 cancer cell line. And as I have mentioned before, we have our research question. Is Rhizophora maculaata bark extract also contain phytochemicals and exhibit pharmacological activities? The answer is yes. And our major conclusion is that Rhizophora mucronata bark extract contains phytochemicals with promising pharmacological activities suggesting its effectivity as a future source of antibacterial, antioxidant, and anti-angiogenic activities, anti-angiogenic agents rather. And for our future insights or recommendation, we recommend further purification, characterization, and elucidation of the phytochemicals in the bark extract. Since we did not isolate or purify, we just determine the presence and absence of the phytochemicals in our bark extract. Next is to further assess the toxicity through in vivo, oral, or chronic effect tests. Third is to further evaluate the toxicity against other types of cancer cell lines. And fourth is to build new and effective environmental conventions through local and national cooperation with the aim to protect Philippine mangrove populations. Since I still have the time, I would like to take this opportunity to thank the DOST PCHRD for their financial support. If it weren't for you, we would not be able to accomplish all of our research objectives. Now, at this moment, I am ready to answer all of your questions. Muli, maraming salamat po.
All right, maraming salamat, Joshua. The community of Southern Luzon State University is very proud of you, as we can see from their post in our Facebook Live. All right, so good job on your presentation and very interesting study that you have presented. All right, so for this round of the Q&A session, we will have Dr. Rosana and Dr. Arrollado to ask Joshua. We will have first Sir Albert. Sir Albert, your questions, please.
Good afternoon, sir.
Hello, good afternoon. I'm Joshua. Thank you very much for a very, strategic work on drug discovery, capitalizing on Region 4's availability of these mangroves. In line with that comment, can you explain to us what is the relative density or population from an ecological standpoint of this particular mangrove species in the country?
Okay, sir. Our country is one of the biggest, or we have one of the largest mangrove area, and Quezon Province is the site of the Rhizophora mucronata. However, this species is very rampant here in Quezon Province, specifically here in Lucena City. It is actually located nearby a small community near a Bajau community.
Okay, sounds good. My next question is, looking at the tables that you presented, you basically tested 3 concentrations of your extracts, your ethanolic extracts, against the different microbial pathogens that you have. There doesn't seem to be a dose-dependent response that I can see, meaning the lowest concentration seem to have a better inhibition or a better anti-angiogenic activity compared to the middle concentration, or in fact, the highest concentration is the most effective. So when you try graphing those values, it seemed to have a bimodal response, meaning there are the extremes will provide you relatively high concentration compared to the middle one. Why do you think that is the case?
Hello, sir.
Can you hear me?
Can you repeat, sir, the question? I'm sorry.
So those dependent assays, you have 3 concentrations: the 100,000 ppm, followed by the 1,000, and then you have 100. I'm just puzzled, why is it there is no linear response of the different assays that you've tested? Meaning, yung mababang concentration has a relatively high response or inhibition, and then the highest concentration has the highest inhibitory activity, and yet the middle one somehow suffered. Why do you think that is the case?
Probably because we have this the maximum effective concentration, and this maximum effective concentration is that concentration at which a uncertain extract can possess or can exhibit biological activity. And in this specific bioassay, the extract at low concentration exhibited also high inhibition, probably because of
Maybe I can help you on this.
Yes,
Sometimes, if you have a very concentrated extract, there are masking effects that may happen that as soon as you have it fairly diluted, the activity may not be sequestered or quenched. So maybe that's why your lower concentration is having an unforeseen or unexpected result. Having said that, I'll have one last question for you. I've noticed that you are quite smart to choose the different pathogens. They're quite relevant, actually, from the context of the World Health Organization. So you've chosen members of the escape pathogen. So that includes your Staphylococcus aureus, your E. coli, your Enterobacter aerogenes. But your results suggest that the extract of this Rhizogram muscronata is effective only against gram-negative. Why do you think that is the case?
Okay.
And this is relevant because there's only a few drug candidates that's actually moving into the pipeline against gram-negative infections. So why do you think your extract may have this delineation in targeting the groups of gram-positive and the gram-negative?
Okay, sir. So as you can see in my results, there was no inhibition against the Bacillus coagulans and Bacillus subtilis. So one probable reason might be their ability to employ the RPPs, or the ribosomal protection proteins, which is a important tactic of many Bacillus species that promote resistance to antibiotics. So probably that's the reason, the RPPs, or the ribosomal protection proteins, sir.
Thank you. That's fairly mechanistic, Joshua. I'm okay with my questions.
All right, thank you, Sir Albert. I would like to correct myself. It will be Dr. Villarreal who's gonna throw questions to Joshua, not Dr. Arrollado. So, Ma'am Myra, can you now have the questions for Joshua?
Thank you, Dr. Malabad. Again, congratulations, Joshua. I mean, getting this far, being one of the finalists, you're already a winner. So I have some questions. The first one is about the choice of your plant species as well as the sample. So I want to know what you think is what makes your study sample unique, because Rhizophora muconata is not unique to the Philippines, and there are related studies done by Indian scientists which are very similar to what you've done, and they also used bark, and in others they used the leaves. So could you tell me in a few sentences what you think are the unique features of your study?
Rhizophora mucronata, based on our readings, Rhizophora mucronata is a poorly studied species of mangrove in the Philippines in terms of its biological activity, because most of the studies here in our country focus on their diversity, species richness, and so we wanted to know if this specific or certain species of Rhizophora macronata can also be an effective potential source of medicines for future drug developments.
Okay, so maybe you need to read some more because the same species have been used by Indian scientists and other countries with the same same plants, the same mangrove species. So maybe you have to choose something, or maybe add something to the effect that it's the samples were specifically the species that thrive in the Philippines. So my next question is, could you tell me more about the MTT assay that is included in your study? So can you tell me about the cells and your treatment time and the number of cells that you seeded in per well, for example, and the doubling time, etc.
Okay, ma'am. So MTT assay is a rapid parametric assay where the normal and
sorry, you don't have to explain the principles. I want the details of your experiment. So I want to know your treatment time and your the observations that you performed. So more on the results rather than the principle. I think everyone knows what MTT assay is, and I've done thousands of them already. Okay.
Okay, ma'am. So the result of the cytotoxic assay revealed that Rhizophora macronata had no inhibitory action, or it had no strong cytotoxic effect against the normal cell line. However, it had...
Okay, so can you tell me the treatment time and the concentrations that you used for your... How long did you treat your cells with your samples and the concentration? So more on time and treatment, the treatment time and dosage.
Okay. The concentration, the highest concentration is 10 parts per million, and the lowest is 0.78. But actually, ma'am, to be honest, we just requested for a test service for this specific, since it is just a further assessment of the results of the anti-angiogenic activity. We wanted to know if it has a, it is to substantiate the result of the anti-angiogenic activity.
So you mean this part of your study was actually done by someone else, because it's not in your thesis. It's not who performed the experiment. So I assumed that you did the experiment. So you had someone, could you tell us who did the MTT assay?
The University of the Philippines Diliman Mammalian Cell Laboratory.
Okay. So the photos that you're the one who included the photos, right? Okay, let me just double check my what I'm talking about. So you included a photo of the cell, is that right? Or it was not you? Sorry, I got confused after reading so many. So I forgot which one is which. Okay, so anyway, you did not include the photo, right, of the cells. Okay, so the next thing is why your choice of the bark is not sustainable. Okay, so you already mentioned that the mangrove species are already under threat by using them as fuel, etc. And you are promoting the use of the bark, which could also pose some harm to the plants. So why did you choose the bark instead of the leaves?
Because we can extract more compounds in the bark than the leaves, and the leaves contains more water, so the moisture content is much harder to reduce during the extraction. And yes, ma'am. And during the collection, all the injured trees were replaced by new trees or mangrove seeds.
So what you're saying is every time you collect the sample for medicinal purpose, you're killing a mangrove tree.
Actually, ma'am, they are not being killed. They are not killed. They were only injured. And the time when we visited it again, they are now healthy. They are now healthy, and they are not dead.
Okay. So one last question. I think they had me warned. Okay. So in your test, you mentioned abnormal thinning of the blood vessels. as one of your results. So how will you validate that? So theoretically, I know you have done your experiments and you've graduated. So theoretically, how would you validate that result to ensure that normal cells or area where which is not threatened by this cancerous mass, for example, will not be affected? Because thinning of the blood vessels can also be harmful to normal, tissues. So how would you validate this result?
There was an abnormal blood thinning, which is an indication of a moderate anti-angiogenesis. So it's meaning to say the bark extract cut off the nutrient and inhibit the flow of oxygen supply to tumor cells, and inhibiting the fine branching of the blood vessels.
Okay, thank you very much. I think my time is up. Congratulations.
Thank you very much, Ma'am Myra and Sir Albert, for those very insightful questions, and also to Joshua for answering those questions. Maraming salamat. So that's John Joshua Belen from Southern Luzon State University, our representative from Luzon. All right, now you can relax, Joshua. Now we are down to the last 3 of our finalists for this afternoon's presentation. It's very interesting, no? It's like we're watching thesis defense part 2 of our students who have graduated. All right, so those 10 minutes could be the longest 10 minutes of their lives. Okay, so but good job to Jason, to Gerson, and to Joshua. Okay, now let's proceed to our next presenter for this afternoon. We have Ivy Grace M. Natavio from the University of Southern Mindanao. So now we're back to Mindanao. Ivy Grace was born and raised in Davao City, but she went to Cabacan, Cotabato to pursue her goals. She was a very active student leader in her college and in her society. Ivy Grace just graduated with a bachelor's degree in chemistry from the University of Southern Mindanao. She will now present her completed study entitled Phytochemical Determination and In Vitro Anti-Urolithiatic Activity Evaluation of Tangisang Bayawak Ficus variegata blumei leaf extract. Okay, I think Ivy is now ready to present, right, Ivy?
Yes, sir. Yes, sir.
All right, the floor is now yours. Let's give a virtual round of applause to Ivy Grace Natavio. Go ahead, Ivy.
Good afternoon, evaluators, and to everyone who is watching. I am Ivy Grace M. Natavio from the University of Southern Mindanao, and I will present to you my study under the mentorship of Sir Randy B. Tumakder, entitled Phytochemical Determination and In Vitro Anti-Urolithiatic Activity Evaluation of Tangisang Bayawak Ficus variegata bloom leaf extract. Now, this will be the contents of my study, the introduction, the significance of the study, as well as the objectives. After is the methodology, followed by the results and discussion, and lastly is the conclusion. To begin with, urolithiasis is a condition where stones forms in the kidney or any urinary tract of our body. These stones are usually composed of calcium stones, where most of it are calcium oxalates. The development of these stones is a multi-process, which involves first the nucleation, which is the start formation of crystals. Then after the formation of crystals, this crystal will then grow, which is the growth. And then these crystals will come together and adhere to form large crystals agglomerates, which will cause crystal retention that thereby promote stone formation. However, the treatment for kidney stones can be expensive, and the use of synthetic drugs may have different effects in our body. This is why we usually utilize plants for our medication. Tangisang Bayawak doesn't have a lot of medicinal properties that is published. So this became an urge to use the Tangisang Bayawak leaf extract for the anti-urolithiatic activity, to also start a new understanding in the field of anti-urolithiatic activity of the plants. This is also the goal of our study, to determine the presence of the phytochemicals and to evaluate the in vitro antiuretiatic activity of Tangisang Bayawak leaf extract. To expand, the study aims to determine the presence of flavonoids, phenols, alkaloids, terpenoids, saponins, and sterols. To evaluate the antiuretiatic activity, it is determined by the effect of the leaf extract against the formation, the aggregation, and the growth of the calcium oxalate crystals. By this, the different concentrations are compared to determine which of the following concentrations are significant. To do this, we use the following methods. First, the leaf extract. The leaves was collected, washed, and air dried. After the air drying, the leaves was grinded and was soaked in a 95% ethanol. Then the leaf that was soaked was filtered, where the filtrate was concentrated using a rotary evaporator, and the crude extract was used for the antiuretiatic assays. For our analysis testings, we have the phytochemical analysis, and then we also have the antiuretiatic assays, which has 3 assays: the nucleation assay, which will determine the effect of the leaf extract on the formation; the aggregation assay for the effect on the crystal aggregation; and oxalate depletion assay for the growth of the crystals. We also have the characterization of the preformed crystal as well. Now, that there are 3 different leaf extract concentration that was used, which is 100 microgram per ml, 500 microgram per ml, and 1000 microgram per ml, and 2 controls, which is the blank and the positive control, where we use Tacit, a drug that is also used for kidney stones. Now, for percent inhibition, optical density is used, wherein optical density is also referred as absorbance. This was determined through UVB spectroscopy. The optical density was input in our formula. Percent inhibition equals to 1 minus optical density of the test over optical density of the blank times 100. Now, for our results, for the phytochemical analysis following the method of Guevara et al., the leaf extract shows the presence of flavonoids, phenols, terpenoids, and alkaloids. It was said that flavonoids and terpenoids also help in reducing the crystal size of the calcium oxalates, and flavonoids had also studies that it reduced the number of crystals of the plant. Now, to confirm that the preformed crystals are calcium oxalate, FTIR spectroscopy was tested. We found in our tested crystals spectra in the left side that there is a high absorbance at 1606 and 1314, which corresponds to the stretching vibrations of C double bond O and CO, which could also be found in the structure of calcium oxalate. An infrared spectra also by the NIST chemistry webbook was also used as a basis for the comparison of the tested crystals. Now, to determine the effect of the leaf extract on the formation of crystals, nucleation assay was done. Found in the table is the percent inhibition, which was determined through optical density and the percent size reduction of the different treatments. Our leaf extracts, the 3 different concentration of leaf extracts shows a decreasing percent inhibition, where our 100 microgram per ml shows 13.38% inhibition. Our 500 has 8.269%, and our 1000 microgram per ml has a negative 2.44%. While our positive control shows a high inhibition percentage at 74.88%. Since our leaf extract shows a significant difference with our positive control, it could mean that our leaf extract is not as effective with our positive control in terms of inhibiting the formation of crystals. Now, for our percent size reduction, our leaf extract at 100 microgram per ml shows 85.98%, and our positive control shows 90.70%, wherein these 2 treatments shows no significant difference with each other. It means that our leaf extract may be as effective or comparable in terms of reducing the size of the formed crystals by the calcium oxalate. We could also see there in our SEM images below that our positive control has an oval shape and smoother morphology, maybe because of the potassium citrate that is in the drug, and our leaf extract and blank control has pointy edge dendritic and X-shaped morphology, where our leaf extract has a thinner and smaller crystals, which we could also see in our microscopic analysis that these pictures, letters A, B, and C, these are the leaf extract treatments, show smaller crystals. There are formation of crystals, but the crystals are of smaller sizes compared to what we see in the crystal shown on the pictures below at letter D, which is the blank control. Now, for our aggregation assay, still has a negative, decreasing inhibition percentage, where our 100 microgram per ml leaf extract shows 26.58% inhibition, our 500 microgram per ml has a 8.839%, and our 1000 microgram per ml has a negative 6.855% inhibition, while our positive control has 36.44% inhibition. We could see there in the table that our leaf extract at a concentration of 100 microgram per ml shows no significant difference with our positive control. It means that in terms of inhibition by the crystal aggregates of the calcium oxalate, our leaf extract may be comparable with our positive control. Now, for our percent size reduction, our leaf extract at the concentration of 1000 microgram per ml shows higher percent at 71.12% compared to our positive control, which has a percentage of 43.97%. It means that our leaf extract may have smaller crystal aggregates formed compared to our positive control, which we could also see in our microscopic analysis. The pictures letter A, B, and C are the leaf extract treatments. You could see there that there are calcium oxalate crystal aggregates, but the aggregates formed are of smaller sizes compared to what we see in the pictures letter D and E for our blank and positive control. Now, for our oxalate depletion assay to determine the effect of the leaf extract on the growth of our calcium oxalate, it has still a decreasing inhibition percentage, where our 100 microgram per ml has a negative 62.99%, our 500 microgram per ml has a negative 102.45%, and our 1000 microgram per ml has a negative 283.87%, while our positive control shows 17.25% inhibition. For the size reduction, we have, and our leaf extract still shows the higher percent at 18.75%, while our positive control has 8.79%. We could see in our SEM images that we found more crystals on the leaf extract, which might cause for the negative inhibition of the leaf extract. Since our optical density is also referred as absorbance, there must be a lot of interference in the mixture when treated with leaf extract that affect the optical density of the mixture. We could also see there in our microscopic analysis the calcium oxalates that was formed during the assay. Now, to conclude, our leaf extract shows the presence of flavonoids, phenols, alkaloids, and terpenoids, where it was said that flavonoids and terpenoids are also helpful in reducing the size or the number of calcium oxalate crystals. Now, for the evaluation of the anti-urothiatic activity of the Tangisang Bayawak leaf extract, we found for all assay that there are formation of crystals and there are formation of calcium oxalate aggregates, but this crystal that was formed are of smaller sizes. We found out also that the lowest concentration of the leaf extract, which is the 100 microgram per ml, shows the highest percent inhibition, wherein for our aggregation assay, the leaf extract concentration at 100 microgram per ml shows no significant difference with our positive control, making it comparable with tacit. In terms of percent size reduction, our 1000 microgram per ml shows no significant difference from the positive control, especially for our aggregation and oxalate depletion assay. Our leaf extract at a concentration of 1000 microgram per ml shows a higher percent size reduction compared to our positive control. To conclude, our the formation of small sizes crystals that is treated with leaf extract could also be an advantage, as smaller crystals can be easily excreted or passed out by our urine. It will avoid crystal retention in our urinary tract, or it will also avoid the stabilization of these crystals as the development of the stone. That will be all for my presentation. Thank you.
All right, maraming salamat. Daghang salamat, Ivy Grace, for your presentation. Now let us proceed immediately to our question and answer for this round. We'll have 10 minutes also for this Q&A session, and we will have Dr. Arrollado and Dr. Rosana to ask their questions for you, Ivy Grace. So can we have Dr. Arrollado's questions first, Ma'am Erna?
Okay. Thank you, Ivy Grace, for your presentation. My first question is, because you perform phytochemicals, screening or assay in your sample, and you have identified some phytochemicals present. Now, among these phytochemicals, which of them have anti-urolithic activity and why? What is the mechanism of action of these phytochemicals?
So thank you, Doc, for that question po. During the conduct of the thesis, we found out that the flavonoids and terpenoids have claims that it reduces the size or the numbers of crystals, while polyphenols have claims to cap with calcium ions, so that there will be no or less calcium oxalate formation. However, for the mechanism, the specific mechanism of this extract of these phytochemicals on its effect on the antiuretic activity, we were not able to have proposed mechanisms for it, since we wasn't able to identify which of the following phytochemicals have cap with our crystals. Maybe these phytochemicals have cap with the formation of the crystals, which affects the further growth of the calcium oxalate crystals po.
Okay, thank you. How do you know that the calcium oxalate crystals formed was calcium oxalate monohydrate using FTIR?
So it was found there po that the peaks for C double bond O and CO, as well as the OH bending for the FTIR, it was used to identify that the calcium oxalate was calcium oxalate monohydrate. We also used the spectra that was provided by the NIST for calcium oxalate monohydrate, so that we'll be able to better confirm that the spectra of the tested crystals are calcium oxalate monohydrate po.
Okay, so I will consider the question posted. Why is it that you use tacit as your positive control?
The use of tacit is because of the potassium citrate, wherein there are published articles that uses potassium citrate for kidney stones. That is the reason why tacit was used for the antiuretic activity po.
Okay. And now, how did you prepare the different concentrations of your extract?
The different concentrations po was concentrated, and then the crude extract that was recovered was weighed, so that we could be able to dilute it for the 100, 500, and 1,000 microgram per ml, which was also diluted with ethanol, since our solvent is ethanol po.
Okay, so thank you, Dr. Rosana. You may take over.
Thank you, Dr. Arrollado. Hi, Ivy Grace. I just have two questions here for you. And I think the first one is regarding the the percent inhibition, that particular tables that you have, it shows that as you increase the concentration of your extract, the values of your percent inhibition actually went into the negative range. And looking at the formula that you presented to us, it's actually on a function of like 1 being in reference to the quotient of your 2 optical densities. You kind of alluded this to us that there might be interfering molecules. Of course, as you increase the extracts in your oxalate mixture, what do you think is the mechanism behind that, considering that you have a green extract? That's a hint.
It was found that while the concentration goes higher and then the optical density also goes higher, it means that there is a lot of absorbance, since optical density is also referred as absorbance. So it might be also because of the matrix of the content of the leaf extract, since you have said, sir, that as the concentration increases, it becomes dark, wherein there will be a lot of molecules that would affect the absorbance of the mixture.
What is the wavelength? What is the wavelength that you use to measure the absorbance?
The wavelength that was used for the assays po is 620 nanometer po.
And what do you think is that major plant leaf molecule that absorbs quite well at this 620 nanometer wavelength?
Do you mean the molecule of the leaf extract po, sir?
Yes, exactly. What would be the most logical molecule that is green in color that absorbs really, really well at the 620 nanometer wavelength?
I think po, sir, it's
a green pigment.
Probably chlorophyll.
Exactly. So I think that 620 nanometer may have a shadowing effect in your assay, because as you increase the concentration of your leaf extract, you might be increasing also that shadowing effect, and therefore you're going into the negative values for your percent inhibition. So that's something that you might consider for any future sort of activity, if ever this is something that will be taken care of by a new grad student, a new undergrad student. My second question is on your SEM, or your scanning electron microscopy procedure. So the calcium oxalate may be suspended or maybe in a crystal form. So when you subject this to any form of electron microscopy technique, there is a dehydration procedure via ethanol or HMBS hexamethyl disilazane, which could potentially, in principle, allow for crystallization because you're removing the water shell. Can you comment as to whether the crystals that you have seen in your SEM are actually real, or could they be just artifacts of the actual SEM procedure?
So for our SEM po, since it was tested by a different school, the sample was given to a different school for the test po. We only take the basis on the pictures that was given unto us, wherein we also compared it with the other articles based on how they describe the morphology of the calcium oxalate crystals that they have found on their SEM images as well, dear Dinpo. That's how we characterize our calcium oxalate that was found in our SEM images po.
Maybe in the future, you can consider having an orthogonal assay to support this characterization other than SEM, which is a fairly superficial, it's a surface morphology method in inorganic chem type of or materials chemistry field. Something to consider. So thank you very much, Ivy.
Thank you
Back
po,
to you,
Doc.
Dr. JC.
All right. Thank you, Sir Albert and Ma'am Erna, for your questions. And thank you very much, Ivy Grace, for your presentation and for answering the questions from our judges. Okay, good job. You can now relax and watch our last two finalists for this afternoon's competition. All right. So can we give Ivy Grace a virtual round of applause for her wonderful presentation? All right. Now we are down to our last two finalists for this afternoon's competition. Let me present to you our fifth presenter. We have from the Western Mindanao State University. Ayan. So marami po tayong representatives from Mindanao for this year's competition. So we have Wenzel E. Perian, who recently obtained his Bachelor of Science in Biology, major in Medical Biology. He graduated from West Mindanao State University as summa cum laude. Wow, very good. His research interests include cancer biology, molecular diagnostics, applied phycology, and molecular networking of natural products. Presently, he works as a university research associate at the National Institute of Molecular Biology and Biotechnology at the University of the Philippines, Diliman, Quezon City. And he plans to pursue his MS studies. He will now present his study entitled Anti-Tumor and Anti-Angiogenic Activities of Crude Methanolic Extracts, seaweed extracts of Soliera robusta greville kailin 1932. So, Wenzel, are you ready to present your study? Okay, I think you are. The floor is now yours. Go ahead with your presentation.
Thank you very much. To the esteemed Board of Judges and all the participants and spectators of the 15th PNHRS week, good afternoon. It is an honor to present to you my bachelor's thesis entitled Anti-Tumor and Anti-Angiogenic Activities of the Crude Methanolic Seaweed Extract of Soleria robusta greville kailin. So for the introduction, seaweeds are prolific sources of highly variable bioactive compounds. This is due to their ability to synthesize a variety of primary and secondary metabolites that exhibit a broad spectrum of bioactivity, including anti-cancer properties, among many others. Therefore, seaweeds provide a promising source of natural products that can be employed in the prevention and treatment of various diseases. Now, cancer is a pathological condition characterized by the uncontrolled growth and proliferation of cells due to the alteration of various homeostatic pathways. Hence, inhibiting such mechanisms to persist is a common approach taken in cancer therapy. With that, the tumor microenvironment promotes immune resistance and is crucial in the proliferation and survival of tumor cells. Hence, stimulating an anti-tumor response and overcoming immune-suppressing barriers prevent cancer progression. Furthermore, angiogenesis is the process of forming new blood vessels and plays a role in tumor development. Therefore, using anti-angiogenic compounds to combat angiogenesis induced by cancer prevents cancer tumors from forming their own blood vessels. Soleria robusta is a species of red algae reported to occur in Zamboanga City, Philippines, and is shown to elicit anti-inflammatory and anti-proliferative activities. Several studies have shown the anti-cancer properties of S. robusta. However, the literature is extremely lacking, and other possible mechanisms of S. robusta have never been studied. Hence, the present study is designed to investigate the anti-tumor and anti-angiogenic activity of the Philippines S. robusta seaweeds. So for the objectives of the study, the general objective of the study was to evaluate the potential of S. robusta methanolic extract as an anti-cancer agent through its anti-tumor and anti-angiogenic activities in the MTT and in the DUC-HAM assay, respectively. Specifically, it aimed to attain the following objectives. First is to determine if there is a significant difference in the number of viable cells between the control setup and the experimental setup. Second is to determine which concentrations of S. robusta methanolic extract possess inhibitory effect against A549 cells comparable to that of the positive control. And third is to determine if there is a significant difference in the vascular density of the CAM assay between the control setup and the experimental setup. Now for the significance of the study. The findings of this study can contribute valuable information regarding the use of Solera robusta as an anti-cancer agent. This can supply substantial data regarding its application as natural products that combat cancer. With that, the possible use of S. robusta in the development of anti-cancer drugs can be supported by this study. Furthermore, this study can also provide high insights into the anti-cancer properties of Philippine seaweeds, as most of them are unknown. In a clinical setting, this can prevent cancer progression and reduce the rate of cancer incidence. So proceeding to the methods, for the preparation of the crude extract, 10 kilograms of S. robusta was collected from the coastal waters of Zamboanga City. And after collection, the seaweeds were washed and were air-dried, ground, and stored. Following the method of Vien et al. In 2014, the dried algal matter was extracted with 100% methanol for at least 72 hours and was filtered thrice. Finally, the filtrate was concentrated using a rotary evaporator to produce a dark green viscous oily mass of Solera robusta. Now for the anti-tumor assay research procedure, the anti-tumor activity of SRME was determined using the MTT assay. The MTT cytotoxicity assay performed in this study was conducted following the method of Mossman in 1983 and summarized in the flowchart in Figure 1. For the anti-angiogenic assay research procedure, the DUC-CAM assay was employed in the determination of the anti-angiogenic activity, and it was conducted following a modification of the method described by Luayat et al. In 2018 and what is shown in the flowchart in Figure 2. For the quantification of angiogenesis, the quantification of angiogenesis was conducted following a modification of the well-established method described by Dikisi et al. In 2019. This procedure follows a multi-step imaging processing approach using Adobe Photoshop's ImageJ or Fiji and AngioTool. So basically, the enhancement of the images were done in Adobe Photoshop. Then, following this order, the images were binarized, segmented, and skeletonized in ImageJ. And finally, the image were imported and analyzed in AngioTool. For the statistical analysis, all data were expressed as mean standard deviation. One-way ANOVA was conducted, and 2 key post-hoc tests for data with significant difference was performed. So now heading on to the results. Represented in Figure 3 are the effects of the various treatment concentrations on the mean cell viability of A549 cells. The graph shows that doxorubicin treatment at 0.34 micrograms per milliliter or treatment 9 exhibited the lowest mean percent cell viability, followed by 100 micrograms per milliliter of SRME, 0.78 micrograms per milliliter of SRME, or treatment 8 and treatment 1 respectively. Furthermore, a significant difference was found in the mean percent viability values, and it is highlighted that treatment 1 and treatment 8 of SRME are homogeneous with the treatment concentration of doxorubicin. So to confirm that the dose testing SRME does not follow a sigmoidal trend, data points on the raw absorbance readings per trial were plotted as shown in Figure 4. This was compared to that of the positive control's raw absorbance readings depicted in Figure 5. Based on the trends of the plotted data points of doxorubicin, it was revealed that indeed the dose testing SRME does not conform to a sigmoidal curve. Furthermore, a non-monotonic dose response curve in the data points of SRME was established. So for the percent inhibition, the effects of the various treatment groups on the mean percent inhibition against A549 cells are presented in figure 6. Similarly, treatment 9 exhibited the highest mean percent inhibition, followed by SRME treatments 8 and 1 respectively. Simply put, statistical analyses reveal a significant difference among the treatment groups and extract dilutions of T1 and T8 that are homogeneous with T9. So for the discussion on the anti-tumor assay results, so many literatures have emphasized the promising role of seaweeds as potential sources of natural products that combat cancerous tumors. Results of the MTT assay reveal differences in the sensitivity of A549 cells to the different concentrations of SRME. It was shown, however, that treatment of SRME at 0.78 micrograms per milliliter and 100 micrograms per milliliter exhibit comparable effects to that of the chemotherapeutic drug doxorubicin at 0.34 micrograms per milliliter. The anti-tumor activity of S. robusta in this study therefore corroborates the findings of PN et al. In 2014 on the anti-cancer property activity of the methanolic extract of the seaweed. This study therefore supports that S. robusta methanolic extract possesses cytotoxic effect against cancer cells. And many studies have reported the anti-tumor activities of many red macroalgae against cancer cell lines, including A549 cell lines, as in the author cited. And interestingly, the extract still exerted weak but substantial cytotoxic effect against A549 cells, which has evolved various mechanisms of resistance against cancer cells, against anti-cancer drugs rather. So for the bioactive compounds in S. robusta, preliminary phytochemical screening of SRME in this study revealed the presence of flavonoid in the crude extract. Flavonoids exhibit anti-cancer effects by inhibiting tumor cells through disrupting carcinogenesis and by arresting mitosis during the telophase stage. Some flavonoids can also restrain cancer development by affecting hormone production. And thus, the anti-tumor activity of SRME in this study can be linked to the flavonoid content in the extract. Furthermore, carrageenans have also been shown to inhibit tumor growth as well as phycobiliproteins. So discussing the non-monotonic dose response of SRME, we look into that the IC50 values of SRME against A549 cells, which were initially sought to be determined in this study, were not determined because of the non-sigmoidal nature of the plotted data points. There are more than 1,000 citations in the literature for molecules that exhibit this type of behavior. And the extract inhibits, this means that the extract inhibits cancer proliferation at specific concentrations only, implying that cytotoxicity of seaweed extract against cancer cells vary on the cell line tested. This phenomenon can be linked to the masking effect induced by other more abundant compounds in the crude extract. So for the results on the angiogenesis quantification, figure 7 shows the sample images for the processing and analysis of the angiogenesis quantification from the enhancement, binarization, segmentation, skeletonization, and the analysis in angio tool. So represented in figure 8 are the effects of the SRME on the vascular density, and treatment of celecoxib at a concentration of 20 micrograms per disc yielded the lowest mean percent vascular density. It was revealed also that no significant difference in the mean percent vascular density values in the CAM areas between treatment groups. So to further validate the anti-angiogenic effects of SRME, data on the total vessel length and total number of junctions were also obtained and analyzed. It was similar results were found, and there is no significant difference in the mean total number of vessel length and total number of junctions. So for the discussion on the angiogenesis and anti-angiogenic results, pharmacological strategies have employed anti-angiogenic therapy as an approach to target this process. Furthermore, it was shown that anti-angiogenic effects exerted by SRME on the total vessel length and the total number of junctions in the CAM sample areas are also comparable to that of the positive control. With that, the present study is the first report on the anti-angiogenic property of S. robusta. Many studies have revealed the anti-angiogenic effect of red seaweeds like Gracilaria coronopifolia, which inhibited angiogenesis through AMPK enzyme activation. Carrageenans and flavonoids have also been shown to exhibit anti-angiogenic properties by disrupting VEGF signaling. So for the conclusion and recommendations, in conclusion, the present study demonstrated the potential of S. robusta methanolic extract as an anti-cancer agent through its anti-tumor and anti-angiogenic activities. It was found that the extract was able to attenuate cell viability of A549 cells via MTT assay at concentrations of 0.78 micrograms per milliliter and 100 micrograms per milliliter. Percent inhibition induced by the lowest and highest concentration of SRME was comparable to that of the chemotherapeutic drug, revealing a biphasic dose response. SRME also exhibited anti-angiogenic effects by inhibiting percent vascularity, vessel length, and mean total number of junctions. S. robusta, therefore, can provide a promising source of natural products that with anti-cancer properties. For the recommendation, it is recommended to employ a more selective extraction method to eliminate the suspected masking effect exerted by other factors in the extract. Solvents of varying polarity can also be used to extract the bioactive compounds, and the extract may undergo a purification process to eventually isolate the bioactive compound. Finally, evaluating the anti-cancer potential of S. robusta in combination with chemotherapeutic drugs like doxorubicin can be useful to decrease the toxicity of the latter. So with that, I reached the end of my presentation. Thank you all for listening, and I am now ready to answer your questions.
All right, thank you very much, Wenzel, and good job on your presentation. Your friends from FB Live are cheering on you, okay? So now let us proceed to our question and answer portion for Wenzel's presentation. We will have for this round, Dr. Arrollado and Dr. Arrollado and Dr. Villarreal. But we will have first Ma'am Myra's question. Ma'am Myra, go ahead po.
Thank you, and congratulations, Wenzel, on completing your research, and of
Thank you
course,
so much,
getting the
Doctor.
funding from PCHRD, and another one also being one of the finalists in today's oral competition. So I guess you already guessed what I'll be asking. I'm basically using the same pattern. So the first thing is like what I mentioned with the other finalists, that your study has also been done. Similar studies have been done elsewhere, using the same species and almost the same methodologies, and we understand that as an undergrad you have some limitations, so that's something that's against you. So but I would like to know if you could tell us what do you think are the unique or what makes your study unique, considering that other similar studies have already been done on the particular sample and using the same test organisms as well.
Yes, thank you so much for that question, Dr. Villarreal. So the novelty in my study is that my study was able to found or be the first report on the anti-angiogenic activities of SRME or Solera robusta. This has never been reported before, and so the study is now the first account on the anti-angiogenic property of S. robusta. This is the main highlight of the study, as studies that have tested the cytotoxicity of SRME or Solera robusta in general have never touched on the anti-angiogenic effects of the seaweed. However, other studies have touched on the anti-proliferative effects of the seaweed, and so the study looked into the anti-angiogenic property as a possible mechanism for its anti-cancer properties. That is one of the main novelties in this study.
Thank you. in your sample preparation, and you acknowledge the limitation in your recommendation, you used methanol, which basically extracted everything.
Yes.
But then later you mentioned the role of VGF pathway, VGF signaling, etc. So in most cases, after the general methanol extraction, it's followed by a series of extraction. So which group of compounds are you really after for the effects that you'd like to see?
For the group of compounds, we're particularly interested with polyphenols, particularly with the flavonoids, as they have been shown to elicit this anti-cancer activity. And in the preliminary phytochemical screening, it was revealed that there is indeed a presence of flavonoids in the crude extract. And it also corroborates the findings on the non-monotonicity observed in the results, because flavonoids have also been reported to affect anti-cancer proliferation in a biphasic manner. So with that, we aim to eventually and hopefully isolate these flavonoids and look into the other groups, polyphenol groups in the extract.
So having you explain that, what do you think would be the next solvent to use following your methanol extraction in order to get the flavonoids?
I think for the solvent to be used, we want to use more. Basically, we also want to use other solvents, non-polar and polar solvents, to really isolate all the possible bioactive compounds that are present in the extract. So maybe we hope to use a solvent like acetone or butane or butanol to eventually isolate some of the more delicate compounds found in the crude extract.
I think you need to read more on that.
Thank you. Yes.
Anyway, my next question is about your MTT assay. So you mentioned, I think in your thesis, you mentioned that you want to establish the effect on toxicity, right? And unfortunately, the results that you got were not were not what you expected, and you're not able to calculate for the IC50.
Yes.
So why did you stop at the highest concentration that you used? Because to get the IC50, what we usually do is to get as high concentration as possible until 50% of the cells were obviously dead. So so why did you stop at the, I mean, maybe the best question is, what was your basis in choosing the concentrations that you used for your assay?
Yes. So the protocol that was observed in performing the MTP assay was actually the protocol being done by the Mammalian Cell Culture Laboratory in UP Diliman. So these protocols are actually non-negotiable. So we had to actually follow with the concentrations that they were using. So with that, that is one of the limitations of the cytotoxic testing, the cytotoxicity testing, because we were only limited to the protocols being observed in the institution that we were able to perform the cytotoxicity assay.
So do you mean you were only able to do it once for the MTT assay?
We're able to do it thrice. However,
you didn't need the concentrations.
Yeah. For the concentrations, there were 8 concentrations, and started from 0.78 micrograms to 100 micrograms per milliliter.
Okay. So can I still ask question, or is my time is up? I'm not taking. I'm not.
Yes, you can still ask.
Okay, one last. Okay. So you mentioned that you want to focus on flavonoids, and we have published papers on the use of flavonoids or the effect of flavonoids on cancer. But in your case, using seaweeds, which group of which specific flavonoids do you think which are present in seaweed would be a potential or a potential candidate for anti-cancer effect? And could you tell me more about which specific cancers are you targeting with this compound?
So with the present study, I believe that quercetin is among of the flavonoids that we aim to isolate. And also, quercetin has been shown to affect cancer proliferation in a biphasic dose response. So we are able to correlate this by, we suspect that the flavonoid found in the extract, or some of the flavonoids found in the extract, could be quercetin. And also, that is like one of the main targets to isolate, and also to determine as well how these bioactive compounds would work if they are presented to in the cytotoxicity test in a more purified manner without the masking effect induced by other compounds that could overshadow its effects.
All right. Thank you, Dr. Villarreal and Sir Wenzel. Can we have Dr. Arrollado's question, Ma'am Erna?
Okay, I have a few questions. I think two. Okay, so first, you use celecoxib as your standard in testing for anti-tumor, anti-angiogenic activities. Why did you select celecoxib when in fact the main indication of celecoxib is non-steroidal anti-inflammatory drug, analgesic, antipyretic?
Yes. So silicosib, one of the factors taken into consideration in choosing silicosib as the control was that silicosib is actually a drug whose inhibitory property on angiogenesis has already been established in many studies. Also, silicosib was the most available compound at our disposal, so it was more efficient for us to use silicosib. However, this does not, we did not try to go around it the easier way, as silicosib has also been found already to exhibit anti-angiogenic activities. So silicosib is a valid anti-angiogenic drug in this setup, Doc.
But the mechanism of action of celecoxib is that it inhibits prostaglandin synthesis.
Yes.
And then I researched on the anti-angiogenic activity of celecoxib. It's 75%. No, it's not that 100%. So I don't know if this is a good standard drug for this test. Okay, so another is you use doxorubicin. Doxorubicin is a multi-agent adjuvant chemotherapy. So it is an adjuvant chemotherapy for treatment of women with axillary lymph node involvement following resection of primary breast cancer. So the cell lines that you use is that anti-cancer. What meaning to say for breast cancer?
No, the cell line was actually A549 cell line, which was human lung cancer. And for the mechanistic action or the mechanism of action of doxorubicin, it actually inhibits topoisomerases during which function in the relieving of the torsional strain during DNA replication. So once this topoisomerase is inhibited, DNA replication of cancer cell may cease to persist, and so it ends the cycle right there.
Okay, thank you. So that's all.
Alright, maraming salamat po, Ma'am Erna and Ma'am Myra. And also, Wenzel, maraming salamat. That was a very insightful exchange of questions and answers from our finalists and our judges. Alright, so Wenzel, you can now relax, and Region 9 is very proud of your presentation. Alright,
Thank you so
so
much.
maraming salamat. Now we are down to our last finalist who will present her study. But before that, we would like to appreciate our very engaging audience who have been posting their questions in Zoom and also in Facebook Live. We apologize that we cannot entertain or give a time for you to ask your questions live, but our finalists will be addressing them via email. So just keep your questions coming, and we will send your questions to our finalists so that they'll be able to address your questions. All right? Now, last but definitely not the least, but still from Mindanao, we have Amera Samporna from the Mindanao State University Main Campus, Marawi. Amera recently graduated as magna cum laude with a degree of Bachelor of Science in Biology, major in Animal Biology. She was awarded with the Best Thesis Award in Biology and was also awarded with a distinction of Researcher of the Year. During her undergraduate years, she was able to conduct research studies and was able to present to local and international scientific conferences. She will now present her study entitled Anti-Angiogenicity and Teratogenicity of Melanolepis multiglandulosa stem bark and leaf crude ethanolic extracts in mallard ducks Anas platyrhynchus embryo. Amera, I think you are now ready to present your study, right? Okay, I think you are. Let's all give Amera a virtual round of applause. Go ahead, Amera. The virtual floor is now yours to present your study.
Thank you so much po. Assalamualaikum warahmatullahi wabarakatuh. Greetings of peace to each and everyone. It's such a great privilege to be here right now. I am Amira Samporna, representing the BARMM and the Mindanao State University. Before anything else, I would like to take this opportunity to express my deepest gratitude to the DOST PCHRD. Thank you so much for giving me this rare opportunity. Mabuhay po kayo. And to the BHRCD and the Mindanao State University, Marawi, and to my thesis advisor, Ma'am Bebs, thank you so much po for the endless moral support. Words is not enough to express how I am grateful today. Maraming salamat po sa inyong lahat. I am here now to share to you the findings of my study entitled Anti-Angiogenicity and Teratogenicity of Melanolepis multiglandulosa, stem bark, and leaf curd ethanolic extracts in mallard duck embryos. Next slide. Allow me to have your ears with this 15 minutes presentation. So here is the outline of my presentation. Next slide. Okay. Center of Diseases Control ranked cancer as the second cause of mortality, and cancer has invaded almost all organs of the body as shown in this table. Expensive cancer treatments, however, seem to be privileged to the wealthy. So low-income cancer patients, however, would just be waiting for their end to come without being treated. Thus, this study was conducted to determine if Melanolepis multiglandulosa plus Melanolepis multiglandulosa, commonly known as ALIM, has anti-cancer activity. Next slide. One mechanism to stop cancer is by inhibiting angiogenesis, thus depriving the blood supply to the cancer cell, and therefore the presence of anti-angiogenic activity in M. multiglandulosa implies its anti-cancer activity. While the use of M. multiglandulosa is common in the locality, but it is very important also to establish its safety for use, especially for those pregnant women. Thus, teratogenic activity of this plant was also conducted. So a negative result to teratogenic test would imply safety for human use. Next slide po. Nowadays, researchers are looking for herbal derivative substances that can be used to treat angiogenesis-related disorder. So, M-multiglandulosa is used by traditional healers for the treatments of several diseases such as sore throat, toothaches, and they are also used as sudorific for chest pains, fever, and it can also you also use this as anti-helmintic, constipation, and tuberculosis. Um, this study made use of M. multiglandulosa because this plant is commonly available in the locality and commonly used by the Maranos in the treatment of various diseases, including cancer. So this study wanted to establish its safety for use as a medicinal plant. Next slide, po. With the above rationale in mind, this study was conducted to determine the anti-angiogenic activity and the teratogenic activity of M. multiglandulosa in molar duct embryo as a test system. And to carry out the above general objective, so the following specific objective were done. First is to determine the presence of secondary metabolites and to investigate whether or not the extract of stem bark and leaf curd ethanolic extract of M-multiglandulosa could inhibit blood vessel formation, and also to investigate if the extract could reduce the diameter on and primary and secondary blood vessels, and also to investigate whether or not if if the anti-angiogenic effect is it concentration dependent, or and determine the percent vascularity inhibition of the extract, and to determine the mortality percentage of the aplatyrincus embryos treated with the extract, and also to determine the teratogenicity of stem bark and leaf curd ethanolic extract of M. multiglandulosa in duck embryos. Next slide. In carrying out the above specific objective, this study can provide scientific evidences in the use of M. multiglandulosa in both preventive and therapeutic use against cancer. So first, in terms of pharmaceutical industry, there are one developing and producing and markets the drugs for use as medication. In this case, for the people, this study will provide an additional knowledge on the utilization of the value of the plant. Additionally, determining the teratogenic activity of this plant could also provide useful information about its potential effect on embryo development if consumed during pregnancy. And also, the finding of this study can also be used as a reference to study physiologic processes and other pathological processes. And of course, for the future researchers, so the information that will be gathered in this study will serve as a guide and additional data or reference to those future researchers who will conduct similar study. So further studies are recommended and are of great help to uncover potential sources of alternative medicine. Next slide po. So this study used experimental design in the conduct of anti-angiogenic and teratogenic activity of the test plant to duck embryos. So the study was carried out by first collecting and preparing the ethanolic extract of stem bark and leaves of M. multiglandulosa, as well as the different treatments. And then the phytochemical screening of the plant extract was initially conducted to determine which secondary metabolites were present in the test plant, considering that phytochemical components can vary among the different geographical locations. And then for the anti-angiogenic assessment of the test plant, made use of chorioallantoic membrane vascularity assay that was based on the study of Raga et al. And Rivati et al. 2013 and 2006 to determine the effect of the test plant to the blood vessel formation. So in this study, 350 duck eggs with the same sizes were procured. So the teratogenic effect of the test plant made use of the morphometric analysis of the 16-day-old duck embryos by measuring its body mass index and other gross morphological measurement. And lastly, the appropriate statistical analysis was used to justify the data collected and provide valid answers to the objectives of this study. Okay, moving on to the next slide. Moving on to the results and discussion of this study. So Results of the phytochemical screening show the presence of alkaloids, flavonoids, saponins, steroids, terpenoids, tannins in both leaves and stem bark of M. multiglandulosa. So the presence of these secondary metabolites may attribute to the bioactive components of this test plant. Next slide po. So the Kamasi results show that both the leaf and stem bark ethanolic extract of M. multiglandulosa had reduced the number of blood vessels similar to the anti-angiogenic positive control. So in this study, I used hydrocortisone as a positive control. So an anti-angiogenic activity of the test plant, especially that of stem bark, was also seen in its ability to reduce the diameter of the primary and secondary blood vessels that was even smaller than that of positive control. So the ability of M. multiglandulosa in reducing both the number and diameter of the primary and secondary blood vessels of deaf embryos implies the potential of this plant to also exert inhibitory effect in the blood vessels formation in cancer cells. Thus, M. multiglandulosa is a potential source of anti-cancer drug. Next slide, please. So this is a percent vascularity inhibition. So it was calculated po to evaluate the extent to which the treated groups inhibited the blood vessels. So as you can see there, the majority of the treated groups exhibit a significant reduction in vascularity inhibition when compared to the positive control. So the highest overall percent vascularity inhibition by M. multiglandulosa was recorded at 1 to 2 milligram per cc concentration that was even more effective than the positive control. So this means that this plant has high anti-tumor and anti-cancer potential using CAM assay. Next slide po. So this graph revealed the percent mortality rate among the treated groups. So the T1 20 milligram per cc has the highest recorded mortality rate and with 0% mortality rate for the untreated eggs. Next slide. Here, the specific parameters were included here. So I just summarized the overall result. So data analysis affirmed that the various concentration of the extract were significantly different from each other, but the various concentration of the extract affirmed that they are just in the same effect with the untreated and negative control, which are reference of the study. So it shows that the extract had the same range of morphometric measurements with the untreated embryos as well as those treated with dimethyl sulfoxide, the negative control with regard to the body weight, the eye diameter, and other parameters. Next slide. So, next slide. Okay. So, whereas the duck embryos treated with the hydrocortisone, the positive control show reduction in few morphometric measurements such as the neck, beak, and yes, go ahead. Okay. Next slide. Moving on to the conclusion of this study, so the stem bark and the leaf ethanolic extract of M. multiglandulosa showed the presence of alkaloids, flavonoids, saponins, steroids, terpenoids, and tannins. The ethanolic extract of M. multiglandulosa inhibited blood vessel formation in developing duck embryos as evidenced by the reduced number and diameter of primary and secondary blood vessels and present vascularity inhibition. And then the anti-angiogenic effect of M. multiglandulosa to the developing duck embryos was concentration dependent, and the ethanolic extract of M. multiglandulosa did not cause significant malformation and teratogenic effect on duck embryos except the leaf extract with 20 milligram per cc concentration that caused reduction in the neck length. So next slide po. So, based on the results of this study, so M. multiglandulosa can be further studied as a potential source of anti-angiogenic phytochemicals that can be used for cancer treatment. In addition, the absence of teratogenic effects on duck embryos might qualify these plants for herbal use for both preventive and therapeutic purposes. However, further studies must be done to warrant its safety and effectivity in the treatment and of cancer and other diseases. Next slide po. Okay, thank you once again. Thank you so much, USF, PCHRD, and the MSU main campus for this opportunity and privilege I am now ready to humbly accept your comments and answer your question. Thank you so much.
Alright, dagang salamat, Amera. Maraming salamat sa iyo, and the BARRMM is very proud of you for your presentation and for being part of the final six of this afternoon's competition. Okay, so for this part of the question and answer for Amera, we will have Dr. Rosana and Dr. Villarreal. So can we have the first set of questions from Sir Albert? Go ahead po.
Hello, good afternoon, Amira. Congratulations for being part of the Magic Six for this afternoon's Natural Product Chemistry grantee from PCHRD. Actually, it's more of a clarification question when we read your thesis as well as this presentation. You're defining one of the assay as this teratogenic. And
Yes.
my understanding is that at least in human system, that is essentially a compound that crossed the placental barrier and then gets deposited in the fetus. How is that somehow being modeled in a duck embryo, wherein, of course, it's already outside the chicken, the hen, and therefore there's no constant sort of exposure or bombardment of a potential teratogenic compound. Can you comment on that and maybe explain to us why this model has been established as a teratogenicity assay?
This teratogenicity assay po was based on the previous study po. So, am I audible po?
Yes, go ahead, please, Amira. Yeah, we can hear you.
Yes po. During the experiment po, the treatment po, was applied on the inner shell membrane that could possibly diffuse into the duck embryos. And in my study po, I used 16-day duck embryo because according to the study of Cliff and Stark, they said that during 16th day of incubation, the embryo has developed scales, flows, and firm, and the feeder germs have begun to surround the embryo. And furthermore, also, it has been revealed by Calder and Snartz that morphometric analysis study of the malar duck embryos revealed the overlap in the range of measurements occurred between the day 13 and 15, where the separation by feeder germ character is good.
Okay, so it's more on the short duration by which that embryo developed, and it's usually within a fully developed Anasplatyrhynchus embryo will happen within 28 plus or minus 2 days. So maybe that's the reason. My second question is on the statistical analysis, because I've noticed you have a lot of tables. I don't know, I think
Yes.
I've seen 30 plus tables in your thesis. I'm just wondering, when you establish the data, especially all those morphometric measurements, before doing any of your ANOVA or Tucky test analysis, did you account for the homogeneity of your means? Or I guess my question is, what sort of assumptions do you need to establish first when you're checking for the variability or different significant differences of your data?
Based on my study po, I just we based on the significant differences among the treatments, and I also did separate analysis for the leaves and stem bark as an initial study. So in our case po, we use in our case po the data analysis na we just relied po on the significantly different among the treatments. So the reference of this study is the positive control and the negative control.
Okay, I guess I'll just have one last question here, JC. How prevalent, or is there a large population, large field of this melanolepsis multiglandulosa in your region, or how is it easily accessible? If let's say you're going to repeat this sort of experiments in the future, how do you standardize your preparations if this is going to be some sort of an herbal products type of development?
Yes, without the plant is very common in field po. And it is accessible.
Can you give us a number?
And this to doc.
Can you give us a number? Like maybe is this farmed? Is this part of an agricultural produce or something along that line?
No po, doc. The plant po is accessible in open fields, forested areas, and even the backyard, in even the side road po. And yes, so but the thing is that no study yet was recorded regarding its anti-angiogenic and other activities.
Okay, I think I'm good, JC.
All right,
Thank you,
thank you.
Amira.
Yeah, thank you, Sir Albert. Can we have the questions from Mamyra? Mamyra, go ahead po.
Okay. Again, congratulations, Amira, for being here, being one of the finalists, and already, what do you call, conquering the research world as early as during your undergrad period. So I have several questions. Basically, my questions are almost the same for all the contestants. The first one is, what do you think makes your study unique, or what's the novelty of your study compared to those other researches that have been done regarding your plant sample?
Although M. multiglandulosa, or locally known as alim, has been commonly used by locals for the treatments of various diseases, but the phytochemicals, anti-angiogenicity, and its teratogenicity study is the first one to be conducted in the region. And also po, since this plant po has been used by some Maranos and local community as herbal plant, so as a aspiring biology student, so I want to provide scientific evidence for this plant, yes, and at the same time to give a first-hand information to the community its safety for use, especially when ingested. And also po, the there the test plant, the M. multiglandulosa that was used in this study po, was an initial study in the region, particularly in Marawi, looking at its phytochemical components, anti-angiogenic and teratogenic activity on duck embryos, and the results of the study is new to scientific community in the region. Thank you.
Okay. So my concern again is about the sustainability of your sample. So earlier, Sir Albert already asked you about the abundance of your sample. So, and you mentioned that these are just those that are what's already growing and not farmed by anyone. So it's not sustainable to use the bark. And in all our studies, we use the aerial parts because for that reason, we don't want to kill the plant. So removing the bark is like removing the skin, which is its primary protection from external environment. So, and a plant like yours should be protected. And the use of bark is not sustainable, like I said. So have you compared the composition as far as secondary metabolites are concerned? Have you compared the leaves components versus the bark?
Yes po, but our objective po is not to compare the results of like leaves and the stem bark po.
Yeah, I understand that's not your objective. What I'm driving at is if it's possible, if they contain the same if they have the same composition, then there's no need to use the bark, but rather just focus on the leaves. I think someone mentioned that it's high in water content. Of course, you can dehydrate the sample. So my concern is you said that you compared. So did you find out if they have the same composition as far as secondary metabolites are concerned?
Based on the results po, na there are different phytochemicals between the leaves and bark, but the same in this study po. So
They have the same or not the same composition?
Based on phytochemical screening po, na they were slightly the same, but on the data gathered from CAM assay and teratogenicity assay na they are completely different din po.
Okay, so last question na raw ako. So my question, pwedeng two parts. The first one is, why did you have to use 350 eggs? One of the things that an ethical committee would guard against is using a lot of samples, even if it's just eggs. It's still, I find your 350 a lot. So what's your justification for the use of 350 eggs for your study?
It's because po, this kind of study requires a maximum samples to yield accurate results. And
sorry,
also,
how many samples?
Under public
how many
health
eggs?
service policy po.
How many eggs per treatment is significant? Okay, maybe I'll ask you that. If you have a set of eggs for one group for your experiment, what number would be a significant number of eggs to be considered, for statistical evaluation?
They are all significant.
What's the minimum number of eggs that you can use for one treatment group?
Okay po. 10 eggs po, 10.
How many treatments did you have?
I have 8 treatments po. I also
8, so that's 80. If you have 3 trials, 80 times 3 would be 240, right? So yeah, so maybe you can include that as one of your recommendations, or maybe not you, maybe the person who's going to continue. So I think I have a lot of questions, but I think my time is up. So thank you very much for indulging me.
Thank you. Thank you po.
All right. Maraming salamat po, Ma'am Myra and Sir Albert. And apologies for the time limit that we have for the question and answer portion. And of course, thank you very much, Amera, for answering the questions of our judges. All right, can we give all our 6 finalists a virtual round of applause? They did a very good job in presenting their studies this afternoon, and being on the top 6 is a very great achievement already. All right, so good job to all of you finalists, and of course we are also grateful to all our 3 judges for their expertise and insights in throwing questions to our finalists for this afternoon. All right, we would like to thank everyone for joining us today. Also our audience from Zoom and from Facebook Live. Maraming salamat po for joining us in this year's oral research paper presentation competition. It's been a very fruitful event, and we have gained new knowledge on natural products from the presentations of our finalists. Now, before we end, we would like to recognize and award our finalists their certificates of participation. All right, so first we have for Mr. Jason C. Alcano. Let me read the certificate. The Philippine National Health Research System with the Central Luzon Health Research and Development Consortium, present this certificate of participation to Mr. Jason C. Alcano in grateful recognition of his active participation as one of the finalists in the oral research paper competition for the grantees of the DOST PCHRD Undergraduate Thesis Grant in Natural Products during the 15th PNHRS Week celebration given this 9th day of August 2022 at Clark Marriott Hotel, Clark Freeport Zone, Mabalacat, Pampanga. Signed by the Executive Director of PCHRD, Dr. Jaime C. Montoya, and the Regional Director of DOST3 and the Chair of the Central Luzon Health Research and Development Consortium, Dr. Julius Cesar Sicat. Congratulations and thank you, Jason Alcano. Okay, next we have the certificate of participation for Mr. Gerson Angelo Armesin. Congratulations, Gerson, and thank you for joining our competition this afternoon. Next, we have the certificate of participation awarded to Mr. John Joshua T. Belen. Thank you, Joshua, and congratulations to you. Next, we have the certificate of participation awarded to Ivy Grace M. Natavio. Thank you very much, Ivy, and congratulations to you. We also have the certificate of participation awarded to Mr. Wenzel Perian. Congratulations, Wenzel, and thank you. And lastly, we have the certificate of participation awarded to Miss Amera Samporna. Congratulations once again, Amera, and thank you for joining this year's competition. All right, so congratulations once again to all our finalists for this year's competition. We would also like to express our deepest gratitude to our Board of Judges this afternoon. We will now be awarding them with their certificates of appreciation. All right, we have first for Dr. Erna C. Arrollado. Let me read the contents of the certificate. The Philippine National Health Research System and the Central Luzon Health Research and Development Consortium present this certificate of appreciation to Dr. Erna C. Arrollado for her invaluable contribution as judge in the oral research paper competition for the grantees of the DOST PCHRD Undergraduate Thesis Grant in Natural Products during the 15th PNHRS Week celebration held on August 9, 2022. Given this 9th day of August 2022 at Clark Marriott Hotel, Clark Freeport Zone, Mabalacat, Pampanga. Signed by Dr. Jaime C. Montoya, the Executive Director of PCHRD, and the Lead Coordinator of PNHRS, and by Dr. Julius Cesar Sicat, the Regional Director of DOST Region 3, and the Chair of the Central Luzon Health Research and Development Consortium. Maraming salamat po, Dr. Erna Arrollado.
Thank you too. Thank you very much.
All right, next we have the certificate of appreciation presented to Dr. Albert Remus Rosana. Sir Albert, maraming salamat po for being one of our judges this afternoon.
Yeah, salamat din. Thank you. I enjoy it.
All right. Thank you so much po. And last but not the least, we have the certificate of appreciation presented to Dr. Myra O. Villarreal. Ma'am Myra, maraming salamat for being one of our judges this afternoon po.
The pleasure is mine po. Thank you.
All right, can we give our judges a virtual round of applause from Zoom and also from Facebook Live? We really appreciate your time joining us this afternoon to serve as our Board of Judges. All right, now we will hear a closing message from Ms. Paula Jane A. De Leon, the officer in charge of the PCHRD Institution Development Division.
Ladies and gentlemen, magandang hapon po muli sa inyong lahat. First of all, we would like to commend our 6 finalists. After a rigorous process to screen and evaluate all the funded thesis projects last 2019 and 2021, all of you were selected to represent your institution and region for this year's competition. You should all be proud of yourselves because presenting your completed studies in a limited amount of time to our panel of judges and to a large number of audience is no easy feat. With this, we congratulate you for this excellent achievement. Next, we would also like to extend our gratitude to our esteemed panel of judges, Dr. Arrollado, Dr. Rosana, and Dr. Villarreal. We appreciate all of you for sharing your expertise and for imparting your knowledge and experiences to our finalists and participants for today's event. Lastly, we would like to thank everyone for attending and participating this year's oral research paper presentation competition for the grantees of the DOST PCHRD Undergraduate Thesis Grant in Natural Products. Hosting this yearly event is such an honor for us as it continues to serve as an avenue for our young researchers to nurture their talents and to showcase their innovative works early in their career. We hope that all of you will also join our other PNHRS conference sessions, which will be held virtually from August 10 to 12. As we have always done, PCHRD will continue its commitment to fully support our young Filipino researchers in realizing their potentials and in building their capacities in health research through our programs in the Institution Development Division. Again, from the PCHRD family led by Dr. Jaime Montoya, our Executive Director, we thank you for your participation in this event. Mabuhay ang PNHRS at mabuhay po tayong lahat.
Maraming salamat, Ma'am Paula, for that message. Again, we would like to remind our finalists that the announcement and awarding of the winners will be conducted on Friday, that's August 12, 2022, during the closing ceremony of the 15th PNHRS week. So the next few days will be the most suspenseful days for our finalists as they anticipate who will be the top 3 winners for this year's competition. But as we mentioned, you are all winners already, having been part of the final 6 presenters for this year's research competition. All right? We are inviting our finalists and our audience to please register and attend the different PNHRS week activities from August 10 to 12, as mentioned by Ma'am Paula in her message. For more information, you may visit the 15th PNHRS week website at 15th PNHRS week celebration.dost3.ph. You may also check the Facebook page of the DOST PCHRD for more updates and information. Now, before we officially end the program this afternoon, may we request everyone to please answer our evaluation form by clicking the link in the chat box or by going to the URL or scanning the QR code that is being flashed on your screens. Only participants who have successfully submitted their accomplished evaluation forms will be given an electronic copy of their certificate of participation. All right, so I hope that's clear. And another reminder, please make sure to input your name and email address correctly. The e-certificates will be sent to your emails within 10 working days after the program. Okay, if you didn't receive your e-certificate after 10 working days, please send us an email with your complete name together with a proof of successful submission of the accomplished evaluation form, and kindly send that to dost3rdsmeeting@gmail.com. All right, also please be informed that we will only accept evaluation forms until 7 o'clock this evening. Ayan, so alas 7 po ngayong gabi ang deadline ng submission ng ating evaluation forms. Alright, again, maraming salamat sa ating mga participants, our finalists, our board of judges for participating in this event. We look forward to everyone's participation in the announcement of winners during the closing ceremony of the 15th PNHRS week. Okay, and there you will be able to cheer for your representatives from your regions, especially those who will be bagging awards from this year's competition. Maraming salamat po. Stay safe and have a pleasant afternoon to everybody. God bless everyone. Maraming salamat po sa inyong lahat. And thank you, Ma'am Erna, Sir Albert, and Ma'am Myra, and all our finalists. Congratulations.
Salamat,
Thank you,
salamat,
Doc Jason.
Thank you
Jean.
po.
Salamat. Ang galing mo, Doc Jan.
Nako, maraming salamat po.
Galing mo. Si Ma'am
Very
Myra.
dynamic.
Ang ganda ng boses mo, Ma'am Myra.
Ay,
Very
nako po. Maganda po siguro